It's an evolution party and you're invited. If you want to come and share your evidence for evolution, I invite you to join the stream. If you want to come on and share evidence against evolution, you're also welcome on the stream. So the link is in the chat and it's at the top of the chat. It's also in the description of the video. So I welcome you to come and share your evidence for evolution. I'm trying to do this on a regular basis and give people an opportunity. And my promise to you is that if you come on and you share good evidence for evolution, I will give you the point. And I'm keeping a list of things that people have come on and shared. And so far, everything that has been suggested as evidence for evolution is actually really more evidence against young Earth creation. So I haven't really seen any evidence for evolution yet, but I've seen fossil record evidence that has been brought against young Earth creation. So I'm also fine with that. If people want to come on and share that, that's fine as well. But what I'm really looking for is, I constantly hear that there's just so much evidence for evolution. And then when I look and try to see, is there really evidence for evolution? It is not only not there, the scientific evidence is actually strongly against evolution. So that's what I'm hoping to make people aware of. And, if you disagree with me, please do come on and share your perspective. I promise I'm going to be nice to you. I'm not going to be mean to you. I'm not going to try to trick you or do anything weird. Okay. So you're very welcome on. And I do want to bring up in a few weeks ago, someone gave me evidence against the young Earth creation position from the fossil record by saying that why don't we find pollen below the Devonian period? And so I had originally awarded that point, but I have recently found that actually we do find pollen below the Devonian period. So pollen has been found in the Precambrian, the Roraima supergroup, which is supposed to be like 1.7 to 2 billion years old. Now that is problematic because pollen, even according to how people reacted to the evidence for this, have said that pollen really shouldn't be preserved in that condition for that long. And so the source for this comes from actually an article in the journal Nature from 1966, long time ago. So, this is showing the area where basically the formation in which it was found, a Precambrian formation. So we do find pollen, and that is actually a problem for the evolutionary timeline because according to the evolutionary timeline, angiosperms didn't show up until like 380 million years ago, something like that. And so this is a problem for the evolutionary timeline because if this record of pollen has been there since 1 to 2 billion years ago, then why don't we have any fossils of angiosperms? Now, for the young Earth timeline, this makes perfect sense because we don't see the layers, most of the layers, as happening over long periods of time. We see those as most of the layers of sediments as sediments that were deposited during the flood. And so we would expect that even if at the layer where we might find plants, there might still be pollen that goes down into like watery areas or something like that, where we would be able to find pollen where sediments had covered and there was no plants. So let's see, is anybody going to join? It might be just me tonight, and that's fine because I brought a little something for you. But I just wanted to, again, say I'm taking that off my list that I had given in favor of evolution, and I'm putting it on my list in favor of creation because that is still standing. And no one has really refuted it as far as I can tell in the scientific literature. If you know something I don't, you're welcome to bring it. But so far, I haven't found anything. And, I can always be wrong. There's a lot of information out there. So if you have something that refutes that, then please bring it. But what's really interesting is that when I tried to find out, like, what, what is the issue with this pollen, the answer I got was, well, most scientists say this has to be contamination. And the reasons that it gave that it had to be contamination are all based on the evolutionary timeline. So it's like, well, we think it was contamination because it doesn't fit in with the evolutionary timeline, essentially. So that's problematic because it actually exposes a major problem that I find over and over again when people are like considering evidence for evolution. They start with the assumption that evolution happened, and then they base their findings it's not always the most scientific way of figuring things out. So this pollen thing is a good example of people are saying it's a contamination, but are they saying that based on the observations? No, they're saying that based on their assumption that the pollen shouldn't be there because it shouldn't be there in the evolutionary timeline. Okay, so enough of that. I'm going to bring just a little bit of evidence for evolution tonight. And it's just one simple thing. And it's one of many I haven't counted them, but I'm guessing thousands, maybe even more than that, chicken and egg problems for evolution. So the chicken and egg problem that I'm bringing up tonight is, which came first, the DNA that codes for the helicase or the helicase? Now, this helicase that I'm showing you, this is a protein, and it's a protein found in every living cell. Okay, so every living cell that we know of has to have helicase. And helicase, what it does is it's the protein that unzips the DNA so the DNA can be transcribed. Okay, so it unzips it. So if you've seen the little videos and stuff of DNA unzipping, this is the little machine that unzips the DNA so that it can be copied. Okay, every cell needs this in order to be able to reproduce. Now, which came first, the helicase itself or the DNA that codes for the helicase? Because you can't code for helicase and get a protein built. You can't get helicase to be built without helicase already being present. So in order for helicase to be built, if you already had the DNA for helicase to code for helicase, you would already need helicase to exist so that the DNA could be unzipped. So without helicase, you can't get helicase. Now let's just talk a little bit more about what goes into making a helicase. So I looked this up because, there's not every cell, the helicase is not exactly the same genetic sequence in every helicase. So, but it's in between 300 and 1500 amino acids in this helicase. So 300 to 1500 amino acids had to come together and be put together in just the right way and fold together in just the right way to make this machine. Now, so that's one thing. And then you have, so if you're saying the helicase came first, then you would have to get these 300 to 1500 amino acids to get themselves together and fold in just the right way. And people have done calculations on the probabilities of a protein coming together like that. And it's just astronomical. Okay. It's just by natural processes. Okay. So that's problematic. But let's just say you're trying to build a helicase. Let's just suppose for a minute that somehow, even though we've never seen it, that we didn't need a helicase for the first helicase to be built. Let's just say we started with the DNA and somehow the DNA was able to build a helicase. Well, what would that take? That would take thousands of mutations coordinating together. So a helicase, it would take between, this is just quick things that I looked up. So if I'm wrong, you can tell me. But what I found is that it's approximately 3,000 to 4,600 nucleotides. So in the DNA, you've got these letters that represent, well, we represent them as letters, okay, but they're really chemicals. And so you've got to get 3 to 4,000 of these chemicals to line up in an order that is a code for building the protein, for putting the amino acids together in the right order. So for, let's say you had some previous DNA, I don't know how you have any working DNA without helicase in the first place, but let's just say you've got a little bit of DNA, but you're trying to build helicase by mutation. You've got to get 3,000 to 4,000 mutations to happen in just the right way, in a coordinated way, to be able to code for helicase. And I don't know how mutations would have that kind of foresight, right? To know what needs to be done. Where is the knowledge of what needs to be done? Why does helicase need to be built? Okay, and then how does helicase come out knowing its job, right? Because it's not just that you've got the machine. You've got the machine and it knows what to do and it does it right every time. So that's really difficult to explain by material processes. That's really difficult to explain by mutation and natural selection. So there's really no explanation for how helicase came about, both because of the chicken and egg perspective, and it's just a massive engineering project. So this is the product of a mind. And that's the main point that I'm making through all of this, is that the things that we see in life, these are clearly the product of a mind. And so you are not the product of natural processes. You're not the product of time and chance and a little bit of environmental shaping. You are designed. And so nobody seems to be joining yet. So I'm going to give it another minute and then I'm going to say good night. Thanks for watching. So, hey, Hillary's emails. Good to see you. Thanks for stopping by. You're welcome to come on the stream if you want to. So on the thumbnail for this, I was holding up this book, Darwin Devolves, which I think is a great book. And so this is by Michael Behe, and he really talks about the problem of evolution going in the wrong direction. Now, what does he mean by that? And there's a lot in this book. So, and I read it actually quite a long time ago, so I may not be remembering the bulk of what's in the book. But the main point that I remember is that, there is no mechanism that has been shown to be able to build organisms, the diversity of life, the complexity of life. We don't have a mechanism, an observed mechanism that is able to accomplish that feat. And in fact, what we see is that, yes, there are changes in organisms, but these are largely moving in a direction that's not beneficial for the organism. Now, there are some deleterious mutations, meaning mutations that take away or maybe even you could say slightly harmful, that can have some sort of second order effect that could be temporarily beneficial. And so, but at the same time, it's still overall a degradation of the genome. And so maybe an example would be like one thing that has been promoted as like a good example of evolution is the mutation that causes sickle cell anemia. So sickle cell anemia, it has a second order effect of when you have sickle cell anemia, you are also sort of immune to malaria. And so for that reason, there's a lot of people in places in the world where they have malaria. There's a lot more people with sickle cell anemia because you could survive, right? You might that mutation might help you survive the malaria, but overall sickle cell anemia is not helping you. It might just help you survive, but it's still going to be a problem for your genetics overall. So hello, pigs can fly. Good to see you. Thanks for being here. Do you want to join? It's an open stream, so anybody can join and share evidence for evolution, against evolution. You can come on and just, say your opinion about it. And so you don't have to bring specific evidence. So I'll just give it another minute. And if nobody joins, I'll shut it down. And right now, I have basically 2 lines of evidence going, and I'm not going to show all of it every time because now it's getting kind of long. But basically, the evidence that people have brought me so far all involves the fossil record. And I can tell you when I pull that up, I have awarded some points to more favorable for the evolutionary timeline than for the young Earth timeline. But by and large, you'll see the list that is against the evolutionary timeline is still a lot longer. So the fossil record is really not a good friend to evolution. So, the fossil record can also cause some issues for young Earth creation, but the fossil record is not, what I can see, it is not good evidence for evolution. But that's what people have brought me so far. I'm still waiting for actually positive evidence for evolution. So what is the scientific evidence? And I'm going to tell you guys my next project is somebody asked me how many scientific papers would I need to see that supported evolution before I could accept it. And, I didn't want to saddle myself with 10,000 papers or something like that. Oh, hey. Oh, good. We have some people joining. So, hey, Tyler, welcome.
Hi, how are you
Matt,
doing?
welcome.
Hi, how are you?
Hey.
So
good to see you.
Yeah,
so
it's a
as far
pleasure.
as evolution goes, what is your idea of evolution? How would you define evolution, and why is it false?
Okay, well, there's different things that people say about evolution. The specific definition of evolution that I'm coming against is the idea that we went from chemicals to the diversity of life that we see today through natural processes. And so that's the evolution that I'm coming against. I'm not coming against the idea that organisms can change, and that, a lot of people use that definition of allele changes, in organisms or something like that. I'm fine with the allele changes in organisms.
Right. Well, that is the definition of evolution, is a change in allele frequency in populations by means of herited descent. So in order to say, well, evolution is false, you would have to show that alleles don't exist, which we have plenty of evidence for, or that they don't change frequency in population through herited descent, for which we have abundant examples and evidence for as well. I think what you're talking more about is abiogenesis or something.
Well, I'm also against abiogenesis, but I'll give people the first cell. Okay, I'll grant you the first cell because people who believe in evolution can't handle coupling abiogenesis with it because they know it's just their game is over there. But I'll give you the first cell, and then how do you get to the diversity of life? That's what I'm asking for.
Well, the mechanisms, there's actually 5 mechanisms taught in every, basic biology classes. You have, of course, mutation, natural selection, gene flow, you have non-random mating, and genetic drift, right? So we have, those are the 5 basic ones, but there are mechanisms by which you get a variety of life. So, and we can, this is demonstrable. So, for example, we see Chlamydomonas, which is a single cell algae, and within a very, very short time, not by manipulating the genetics, but only by introducing a predator, it formed into a multi-celled organism, and not a colony, right? A colony, in a colony, a cell can break off, live on its own. In this case, it formed a true multi-cell. It has a membrane that surrounds it, has cells that specialize, and when it reproduces, reproduces as a multi-cell. So that's macro evolution, going from single cell to multi-cell. And this happened, I think, a matter of, a couple of years of experimentation. Perhaps the other gentleman would like to chime in.
Yeah, go ahead and
I
respond.
want you guys to hear me.
Yeah, we can.
Okay. Well, I wanted to point out, so the example, Tyler, that you gave with the algae becoming multicellular is actually not true, and there's actually a couple of examples as to why that's not the case. So when it became supposedly multicellular, what was actually happening is that there was an issue with the dividing of those cells. In other words, they weren't able to fully separate, and therefore they got stuck together. So it's actually a degradation and not a benefit. The only reason it was beneficial is because I forget what it was called, the organism that was coming to attack them was not capable of fully consuming. That was all, right? But ultimately, they lost an ability, not gained one. That's the issue. And it's very likely that that's going to be a continuous pattern amongst all life, right? Just to be clear, we're actually losing a lot of genetic abilities over time per generation. So,
where are you getting that? Because all the
right.
biologists and geneticists, they're
Yeah, they make a lot of assertions right away, and then they end up retracting those statements later. For instance, with the issue with the CIT gene, right, that's provided for the digestion of citrate in the Richard Alinsky experiment, the long-term evolutionary experiment on E. coli. What happened in that situation is that they assumed that because they saw the digestion of citrate, that therefore this is some kind of a new evolutionary pathway. That, of course, now it can actually do something new it was not able to do before. Turns out that wasn't true. It was already able to do that from the beginning, and they just didn't recognize which gene was capable of producing the enzymes that did the metabolic portion of that. The issue was that it was not only promoted in situations where oxygen was not present. So in other words, you had to be in a situation where there was no oxygen present. Basically, it had to be inside of an orange or a lime or something like that, and then it had to basically eat its way out. So it was like a certain situation that would have to be arisen for that to even express. And it was actually a broken gene, so therefore it was actually being expressed constantly without that necessity. But there was actually an issue because it also got rid of its ability to digest fructose and sucrose and a few other different things that were required for normal processing there. And actually, the same daughter genomes that came from that were far degraded and actually died much faster than their predecessors because of this mutation. That's just one example. One more thing that's on top of that is that it lost the ability to use its flagella, which is the outward, basically it's like a little motor, like a small hair-like structure that rotates. Yeah, you know about that. So it lost because they were using a centrifuge to rock those things back and forth. The food was just hitting it right in the face basically the whole time. So it didn't have to use it anymore at that point in time. So when they tried to reintroduce this culture back into the wild, it just died. It had no way, it had to be coddled so that they can try to keep it alive. And that's just another example. So in other words, it's the reverse mechanism of what's being promoted to say this is how we got the things we have. It's actually just throwing out those things in reality is the issue. So that's why I think Rebecca's correct when she's saying, look, it's not necessarily that we're against evolution in the sense that things change over time. That's not the issue. The issue is can the mechanisms that are talking or that we're being told about are actually sufficient to bring about the things that we're talking about. And that turns out to be quite a mysterious issue. Now, one more thing I wanted to point out, and this is just to clarify, there's actually a mechanism called mutator genome decay that's been recognized in the medical research for about 10 years now. If you go to NCBI research website, or if you go to like PNAS or anything else that's going to be pulled for medical documentation through like Google Scholar, just type in that phrase, mutator genome decay. You'll start to see what I'm talking about. There's multiple papers from different experiments that they had done, not just for the E. coli, with multiple different ones. They had the one with the fruit flies where it lost its wings. There's a few different things that popped up there. And there's also ones where they do like the cross verification of genomes between things like, mice. I think, I don't remember what it was. It was like, it's been a minute since I had to go through some of that. So I know that they were trying to like compare different genomes and see what was happening across those genomes over time. And as I was mentioning before, the mutations that were taking place were actually, as she said before, deleterious per generation at a set rate. So we're actually not increasing the genomic information, but we're decreasing it over time. And the problem you should see as an evolutionist is how can your mechanism work if we're always losing that information every single generation? If that's the uniformitarian principle, right? That's the main issue there.
I'm not sold on, if you're talking about genetic entropy, I'm totally not sold on Sanford's idea of that. And as far as, what about myelinase? You got bacteria that developed the ability to
So because there was
which
an
is a
enzyme,
man-made product, it hadn't been here on Earth. I mean, all mutation, if it's a beneficial, it has to be environment specific, right? So you say, oh, well, these cave fish, they lost their eyes, but so it's not gaining anything or whatever. It's not about gaining or losing. It's about adapting to your environment, correct?
no,
So
that's not what I'm
eyes
talking
in a
about.
cave, they rub up against the side of the cave. They
And
get
infected.
infected
Yeah.
easily, so forth. So not having eyes in a totally dark cave is actually beneficial for that for that environment, right?
We agree.
Okay, so you take that fish, put it out into the sunlight, put it in a pond, and now that same mutation that was beneficial is now it's I wouldn't say it's harmful, but it's no longer beneficial. So
They would
maybe
be easy pickings at that point, right? Sitting ducks. So yeah, basically, I mean, I agree with you, right? That there are situations where it is, in quote, beneficial, right? The issue arises because the issue with that right there, that it lost its eyes, the gene remained. They actually could regrow those eyes over time, and that was reversed. They were able to show that it was reversed over time. So I understand where you're coming from, and yes, there is situations where it can be beneficial to, in quote, lose something at a time. But that's the issue, is that can you sustain that? And if you lose the genes for it, is that going to produce the thing you're talking about as far as the main, we're talking about evolution as a whole, driving the production of these things, not the opposite effect. And that's what we're seeing is the opposite.
well, it's not losing the genes. It's that during embryonic development, enzyme and so forth, you got proteins that activate, deactivate, those, they just like you can chickens, you can grow chickens with teeth, for example. But they have the genes for them, they're just not activated.
Yeah, there's a promoter for that,
I
right?
mean,
Exactly.
as an old farm boy, you might hear I've heard the term as rare as a hen's tooth. So
Hmm.
every now and then, you'll have a hen that's born with a tooth. Now, um, if where you're coming from is, well, it was designed perfect in this, I don't know, static way. why would you give hens or chickens the ability to grow teeth if they're never going to use them, right?
Well, how do you know that though? That's a good. I mean, the thing is, the question assumes certain things, right? Automatically, so you have to check your assumptions and see whether or not they're actually valid, right? So we don't know what God's intention was for them to eat certain things. We don't know what they had to deal with back at the beginning, right? That's again, it's an assumption we're making by throwing it into the mix, and then we're asking a question, assuming it's going to actually have relevance in our discussion. But ultimately, we don't know. We weren't there, right? So I
Yeah,
agree with
but
you. There's things we have
right,
questions about for sure.
right. But I mean, are you both young Earth creationists?
I mean, I am personally, but I mean that's irrelevant to the discussion, though.
No, I'm
Me too. But
okay.
I
So,
do want
so
to say, like, people like to bring evidence against young Earth, but this is what I'm seeing over and over again, is that it's much easier to attack young Earth than it is to actually present positive evidence for evolution,
well,
right?
that's weird because that's how I see it. It's much easier to just try to attack and nitpick evolution than to bring evidence for uh, young Earth creationism. That's why I asked.
No, I mean,
Because
I can
because
defend it.
be, because it's first, if you're going to say, well, this is, how it was done, and then if it's a literal reading of the Bible and so forth, and you're claiming that, the God of the Bible did this, to bring that into a scientific area, thing you have to do is show the possibility that such a being exists. I'm not saying that he doesn't, but what I'm saying is you have to show the possibility. And for that, you have to tell me what it is. Is God pure energy? Is the fields of some kind? How do you quantify it?
Can I
And
ask you something?
what are its units, right?
Wait, let me just say something real quick, Matt. Tyler, but see, I don't know about what Matt is going to say about this, but for me, I say, look, I believe a story. Okay. I believe the story that in the beginning, God created the heavens and the earth. That's a story I believe. I admit that that's a story. And
Okay.
I can't offer a scientific explanation for how God speaking was able to bring into existence all of these things. That's not scientifically testable. But the reason I'm nitpicking and attacking evolution is because evolution is also a religious story, but it's being promoted as science. So if it is scientific, if it's not just a story, it should stand up to scientific scrutiny. And it does not. It has been
Well,
contradicted over and over again. So
well,
I'm
I mean,
asking you to give the scientific evidence that evolution is true.
well, I mean, you could go into ERVs, for example, endogenous retroviruses.
Yeah, I know what they are.
Okay. Well, she may not. I'm not sure.
I
Yes,
got
I know all about ERVs. Yes.
Right.
what
So if you had these specially created kinds, then they should not share ERVs.
I
Uh,
disagree with
uh,
that.
uh, I mean, if you have like the first created kind, whatever a kind is, it's not well defined. But from there, you say, okay, you diversify, but they're all, they're actually what you're talking about is more like clades, right? Where you have a clade, and then the descendants, all the descendants, they vary, they diversify from that clade. But you would get back to a certain point where this clade A has nothing to do with clade B, which has nothing to do with clade C. And when we look at the constrained areas of the genome, that's not the hierarchy we see. We see a nested hierarchy. And
Mhm.
if chimpanzees are its own clade, and human beings are its own clade, then there's no way that they should be sharing in ERVs.
I disagree with you, and I'm going to explain to you why. So first and foremost, so you guys, as far as evolution is concerned, you always have to assume that there's a bottom-up scenario where there's some original organism that had to branch off into multiple things. That's why you have the way you expressed it to me in the first place. Now, with the creation model, we assume that God already had genetics that were already diversified, already had the things that were needed there for all the organisms, depending on what they were going to be doing, right? From the start. So everything's already preset and preloaded. That doesn't mean that they don't share genes. It just means that they have genes that are specified to them, and then there's some genes that may or may not work for them in certain situations. So because we don't know what every single organismal trait might be. We have no idea. That's what we're actually investigating. That's the whole point of science, is to try to investigate that sort of thing in biology. Now, if it comes to things like ERVs, think about this. We actually know that viruses at large are very beneficial to us. There are things that we can't do without viruses, in fact. We can't grow a placenta. We can't even see. Our eyesight doesn't even come into us to us until we actually have a virus that infects our eyes. There's all kinds of things that come with viruses. Now, ERVs are what I would believe to be original viruses that actually had intended purposes from the start. And if there's certain segments of DNA they're supposed to affect from the beginning, and we have shared, in quote, genetics regarding certain abilities there from the start, that is something like a primate, it would only affect that portion of the DNA. So the very likelihood that it would be affected there is very high as opposed to not. Now, that's again, this is an assumption. I'm not saying that you have to believe that. But just to be clear, it does make sense if you take it in light of what I'm talking about. And I can understand where you're coming from. If I was to assume that everything had to be from the bottom up, then yes, it would make sense that this is another way you could do it. However, that's assuming that there is no mutated genome decay that I mentioned before. And again, if you don't believe in, like I said, genetic entropy, like you mentioned, you have to kind of take what the NIH is talking about. I didn't say this. This is something that the NIH itself is promoting and talking about. I mean, you'd have to go against the entire medical industry if that's the case. So I don't try to promote something just because,
As far as I know, the genetic entropy does not have high support, much less scientific consensus.
It doesn't have to. It does not have to. But just to be clear, the medical industry actually relies on this kind of information. That's why they've been writing about it for 10 years now. And that's actually, like I said, they would be publishing these papers about it unless it actually was a real mechanism.
Well, I'll leave that for the experts to decide.
Okay, no, I agree with you.
But all I know, it doesn't. Now, if it gains scientific consensus, then I'll change my mind.
Well, I have a question, and I mean, you've been very cordial and very kind. You're not like the average person I'm used to dealing with, just to be clear, and so I really appreciate that. But I mean, if for say it was actually true that the scientific consensus was against the creationist hypothesis simply because they view it as religious instead of actually true because of evidence, why would you think that that's something that that we as creationists should have to worry about? Like, if we go against that, is that something that you think is really an issue?
I'm not following your question.
If we as creationists do not agree that consensus is actually important when it comes down to what is true, is that actually an issue?
No, I mean, that's your right to believe as you want to. The only problem I have really with religion is when it starts to impede and encroach on my life. But aside from that, I mean, who am I to tell somebody else, what to believe or not to believe?
No, certainly. I don't think it was. I didn't mean it to be taken that way. I'm sorry. What I meant is like the actual logic of it, right? So, is it not fallacious that if some group is telling you this is true, but you didn't actually get to verify it yourself, therefore that is, not fallacious? Is that correct? You're saying that
I'm
because there's a
sorry.
large
I'm sorry. I'm still not
Let
consensus.
me
following this
just
at
jump
all.
in and say something real quick, because I
Yeah.
think he's getting confused by what you're saying. But you're basically saying it doesn't really matter what the consensus says. The consensus can be wrong, right? Like, is this
That's
the point
exactly
you're?
right.
And so actually, that's my question for you, Tyler, is, if you look up just all the times when the majority of scientists believe something and then it turned out to be wrong,
Mhm.
you'll find a very long list.
Right.
And
Well,
in fact, if you just read the headlines this week, they will overturn stuff that a few weeks ago they were saying this is absolute, we know this, and then it's overturned. Okay.
right, right.
So
But let me put it to you this way. That is actually the strength of science. When we first coined the term science, were you expecting all the scientists to say, okay, this is what we know about the earth, about the universe, and everything, the natural world, and we got it right the first time, and there's no reason to explore, or I mean, that would be ridiculous, right?
oh yeah.
The whole purpose of science is, it's a self-correcting process, correct?
And
So,
yeah, I love that.
I do agree
I
with
love
you.
that. And
Um,
what I'm advocating for is the taking the dogmatism out of it. I'm fine with us overturning things that we previously thought, but there's a dogmatism that comes. But I want to ask you, because this is so, you see, for a lot of people out there, they're not interested in science at all. And so they're kind of taking a good strategy by just saying, I'll go with the scientific consensus because I don't want to look into it myself. But you actually seem like somebody who is interested in science. Seems like you've looked into this a good bit. So for someone who's interested in science, what's making you choose to just go along with the scientific consensus rather than just being part of like discovering the new, like the truth, the things that we need to overturn?
right, and when you say the truth, I assume you mean the creationist truth.
No, I mean
no,
whatever
she
the
just
truth
means like in
happens to
general.
be. Whatever the truth happens to be.
Oh
If the
yeah, I'm
truth
all for
happens,
well, I'm all for the truth in general. And if somebody says, oh, shows me to be wrong in a certain area, then I'll be grateful because now I don't have to be wrong about that anymore. But as far as why wouldn't I accept, for example, the creationist viewpoint and so forth? Well, one thing, I would expect that the Bible would also um, the scientific discoveries would then correspond with what the Bible says, right? And then you got things like Noah's flood, and I don't know if you think that that was a literal event, but the geology. I mean, you look at. I mean, you got people. Maybe you don't agree with them, but people like Kent Hovind, who says, "Oh, all the layers were created in 1 year," or something like that. And you have Aeolian sandstone, which is formed in deserts. It's basically a sand dune, and you can tell that not only by the angle of the sand, but also by the frosting. When the sand gets blown around, it gets tiny little scratches on it and gives it a white frosted look. You only get that in Aeolian sandstone. You water it, you have something, you pour water on it, it gets dark, right? So you cannot have Aeolian sandstone sandwiched between shale and limestone, for example, and say it all happened practically at the same time. That had to, and we've looked at, how long it takes limestone to form, how long it takes Aeolian sandstone to form and so forth.
Can I
And
ask you a question?
it's not going to happen in 6,000 years. Yeah, sure, go
You
ahead.
know, I would agree with you that if we're just assuming that there's natural process involved in making those, that they would, maybe take longer in some situations. However, there's a lot of assumption there. Are we not taking a moment to just think about what, I mean, assuming from the beginning was there, right? So if you're assuming there was already just regular sand, we have no idea if there was already the Aeolian sand that you're talking about to begin with. We don't know, right? We weren't there. So for us to make that assumption, we actually have to know what was there, or at least admit that we have an assumption there, right? You see what I'm saying? This is something that Dr. Rob Stadler talks about in his book. It's called The Scientific Approach to Evolution.
Mhm.
We have to be able to see what we're assuming beforehand. Now, that isn't to say you could be wrong. I'm just trying to clarify. We just want to make sure we tell people, hey, this is what we're assuming from the start in case we do have something wrong, right? And then from that point, we can try to critique and see if there's anything that could be different, if there's anything that could mess up our particular theory, right? And that's for everybody. It's not just for, it's for anything in science, just in general, right? But that's
Right.
actually one of the reasons why he's doing the book in the first place, because he started to notice that there were a lot of, quotations and statements that just didn't actually comport with the data. And he said, well, if you start looking at what they're assuming, you start to see why a lot of times. A lot of it has to do with bias and assumption. That isn't to say that they have everything wrong. It's just to say that they are taking a skewed look or like a lens, per se, and they're assuming things that maybe don't have to be there, right? But that's for anybody, just to be clear. So I'm not against you for that. I think that you're doing a good job trying to clarify. But just want to let you know where I was coming from.
Okay. And when you, for example, because there's a lot of very religious people that accept the Big Bang, they accept the evolution and so forth, and they say, well, I believe because of faith or whatever. I have nothing to say to them. I mean,
Yeah, I
it's
get
only
you.
when, but I mean, that's their prerogative. But when we have people trying to get things put into or taken out of the public school education, not because they can show a better scientific model, but only because they feel threatened by the current model. If you're going to it's perfectly okay to go and say, I'm going to challenge evolution. I'm going to challenge the Big Bang. But, when you're talking about science, you better bring it. You can't say, well, I feel this way, or, or this is what I believe. That holds no water, right?
No, I agree with you.
And
Hey,
Totally agree.
I'm
Totally
here for the week.
agree.
right.
Yeah, I absolutely agree with you.
So some people, they say, oh, the Big Bang theory, for example, it doesn't, they don't like it. Although, the one who came up, Lemaitre, he was a Catholic priest, and
Exactly.
the Pope seemed to like the idea very much, but some people do not. Right now, if you're going to look at, say, I don't know if you agree with the Big Bang theory or not, but uh, when Gamow, Alpharin, and so forth, these were scientists like in 1948. They reasoned, okay, if the Big Bang happened, if the universe was once in a hot, dense state and has now expanded into its present cool and sparse state, there should be a leftover uh, radiation, the cosmic background radiation. Now, they had no evidence of it at the time, but just, using physics and mathematics, they, they came up with, okay, this should be its temperature. This should be its peak, the peak for different the different energy levels and its overall isotropy and so forth. And it was like, well, in 1964, that's when Penzias and Wilson discovered the Big Bang by accident. But it wasn't until like 1989. So we're talking about over 40 years later when we sent up the Kobe satellite, right? And we got actual readings. And when you look at the data that we got from that Kobe satellite, match it up to the predictions made by Gamow, Alpharin, and Herman in 1948, bam, they knocked that prediction out of the ballpark, right?
It was off
And
by an order of magnitude, actually.
no,
It's still
actually,
really close. But I agree, it's still a good
It
estimation.
was, I mean, it was still very close. Now, here's the thing. You're not, I'm on 10%. You're not going to get that reading. I mean, you would have to, they did make a presumption. You always have to make some type of presumption. The presumption was that physics from the beginning of the initial expansion were the same as the physics we have now. If they had been wrong, then the graphs, the data, and the predictions should be, shouldn't match up whatsoever. If physics has changed, since the beginning of the initial expansion.
Right. I
So,
mean, we can go a number of different ways with that,
well,
because that also gets into like metaphysics and
the
stuff like
only
that.
the only way you're going to have to, the only way you can say, well, they were wrong or whatever, you're going to, you're going to have to show where they got the mathematics wrong, where they got the physics wrong.
Okay, I can tell you where. Let me interrupt here, okay? Because let me just say, Tyler, I like that you're bringing evidence, but I think you need to go back and look at what's going on with the Big Bang right now, because the Big Bang is actually in major trouble right now. And what has been done over the years to prop up the Big Bang is whenever something doesn't fit the model, they invent an entity. Okay, so right now we've got 95% of the universe is completely unobservable. Dark matter, dark energy. And why did we come up with all of this stuff? To prop up the model that doesn't work. Why did we come up with inflation? Because without inflation, which is just a ridiculous idea that has no observations that match it, why did we come up with that? Because we needed it to save the Big Bang from three problems. The cosmic microwave background, you brought that up as evidence. They were off by an order of magnitude. Oh, surprise. We see radiation in the universe. Wow, what a surprise. There's lots of radiating bodies in the universe. And it turns out it's not what it should be if it is from the edge of the universe. Because what we see when we look at the cosmic microwave background, there's a buildup where it's along the elliptical plane of our solar system. Now, if this is really coming from way beyond our solar system, it's very unlikely that we would actually have this, anomaly that lines up exactly with our elliptical plane. If you haven't looked it up, just look up the axis of evil. It's a problem for cosmic microwave background. And there's so many problems with the Big Bang. We could have a huge discussion about that.
Well, I don't want to be disrespectful. It sounds more like conspiracy theory type things.
Well, I mean, just to be clear, though, I mean, conspiracy theories start off that way and oftentimes end up being shown right, and more often than not. Just to be clear, but I agree with you. We shouldn't just throw random ideas out there if we don't know everything. She sounds like to me she's actually done her research, and for the record, I remember seeing her do a couple of debates about this, so I think she's actually done a lot more research in general than the average person.
I'm getting from secular sources. I am not getting any of this from a creationist source. Not one thing that I said is from a creationist source. You can look it all up right now. You can look it up, do AI and find the sources. Everything that I'm telling you is true. The Big Bang has a galaxy formation problem. In fact, I just saw in the popular media, they're ready to double the age of the universe. Now, I haven't, and last year, somebody came out with a paper about that saying, hey, to fix this galaxy problem with the Big Bang.
The James Webb telescope findings. Yeah, I remember seeing
exactly. Look, there are so many problems with the Big Bang right now, Tyler, and you can find all of this in the secular literature. All the problems I'm presenting, none of this is coming from a creationist source. Look up axis of evil, look up galaxy formation problem, look at the dark energy. Where did they get the dark energy calculation? Read Adam Rees's Nobel Prize lecture and see where he got the why we came up with dark energy. It was because he calculated based on the expanding universe. He calculated the supernova based on the expanding universe model, and he came up with a negative mass for the universe. Now, instead of just questioning the model, instead, now we have this invented entity, dark energy, where we have no observations for it. We have no idea what this could be or where it came from, why it suddenly started accelerating the expansion of the universe. It's just to save the model.
Look, if you know, like I said, it's always okay to challenge a model, but you have to have a better model to replace it with. So until
I
you
actually
do,
disagree with that.
until scientists have a better model, I'm not saying it's not okay to question it, but to say I know that it's wrong, or I think that's a step too far. Now
I would
there's
agree.
We don't
and,
need we don't have to have a replacement model. If something is not true and we can show that based on our empirical observations, we should stop
in
believing.
science,
We should
in
stop
science,
promoting it.
in science, yeah, I mean, you can have a model, you can have a theory. That theory can always be updated. There can be new evidence that comes in, right? Okay, well, now we have new evidence, that gives us a better understanding of how the universe works and so forth. I don't see any of it going back to like a 6,000-year-old universe.
Can I point you to
I
some
mean,
of that?
you're just I mean, and like I said, if you're going to say no, it's like this, then the Big Bang is wrong and these guys are wrong, and that, then you're going to have to show where they got the math wrong. You're going to have to,
Right.
okay, here's the formulas they use, here's the math, and here's the real math, and
It's
here's
already
how
been done.
it's
Yeah, I mean,
if you look over at okay, you calculate the age of the Earth, I mean not the Earth, the universe, 1 over the Hubble constant. It gives us about 13.7 billion years old. Now, though there's a lot of parameters, data that goes into that formula, and you can update that, but
well,
you'll have to
you
show
understand that they're using an
where
assumption
one
of
over
how
the
fast light travels and things like that, right?
yeah, it's v equals h naught times d, and then the the recessional velocity equals the Hubble constant times distance. Um, but but anyway, all I'm saying is you have to show, you have to give the real math. That gives us the answer of 6,000 instead of 13.8 billion, because now
Right.
you're saying one of us is off by a factor of 2.3 million. That's quite
Yeah.
that's quite a lot, right?
So Tyler, have you
Um,
heard of Dr. Russ Humphreys? Have you heard of him?
no, I don't think so.
So he's he's a geologist by trade and PhD, of course, and he actually went into electromagnetism. That was his main field, and he started calculating the electromagnetic decay rates for all fields in the galaxy, or at least or the solar system, I should say. And so he actually calculated that for, I think it was Jupiter, Neptune, and a few other planets there, including ours and the moon. And he was able to accurately depict those using a creationist model. So assuming the 6,000-year-old model, and assuming that everything was formed by water, to within the same order of magnitude detect exactly what was going to be, or to depict what was going to be the active decay of that. I'm sorry, what the actual the decayed electromagnetic field was for each of those planets. Just to be clear, so each of those heavenly bodies was being accurately depicted by his model alone compared to his secular counterparts. And it was not found out until the Voyager 2 went out on its mission out there to do those verifications. But that was many years after his predictions. So just to be clear, he made a very accurate depiction or prediction about what was going to take place. And it's not promoted. It's not shown in any textbooks or anything like that. Nobody talks about it because, again, you never even heard his name, right?
Well, No, I mean, if he published it, I should be able to see it in Google Scholar, for example.
Yeah, just look up Russ
I don't know
Humphreys.
if Russ Humphreys. So, and there would be, one thing about science, you make you publish an article, and if your findings are strongly go against, what the consensus is, everybody, in science, the first thing they're going to do is try to prove you wrong. And when they
Yeah.
cannot prove you wrong, then it becomes accepted. And you say, okay, this, the guy was right. We did we did the math and we've done it over. We've checked it. We've tried to disprove this thing left and right, which by the way, I know you don't believe it, but evolution would fit into that category. People have tried to disprove it left and right. When you cannot disprove it, and remember, in science, nothing is ever proven. The proof is for mathematics. In science, there's never an end, a conclusion that this is how it happened. It's always open. The open inquiry, peer review, that's what gives science its strength.
Well, I don't know
And
if you heard about the
real
peer
quick,
review issue. Yeah, go ahead, Tyler.
I'm just saying I'm like on 4%, so I may lose you soon.
Well,
But
let's just say, Tyler, I want to just jump in real quick then, in case we lose you. I want to thank you for coming on. And it seems like you're a very knowledgeable person, and I appreciate you being willing to come on and talk to us. So thank you, and I hope you'll come on again.
Yeah, it was a great conversation.
Yeah, thank you for having me on. And just let me leave you with this. If one thing that with the creationists, and I don't know if this is your viewpoint, but a lot of the creationist viewpoint is that the reason we can trust the Bible narrative is because the Bible is infallible. That is, it is inerrant. And I could put it into a syllogy like, if God exists and he has revealed his divine perfect word to us through scripture, then the Bible is inerrant and true, right? If P, then Q, it follows, right?
Yeah.
Now we look at that inerrancy. We look at, for example, the birth narrative in Matthew and Luke. Matthew has Jesus born during the reign of King Herod the Great. King Herod the Great died in 480. So that Jesus would have been born before. You look, Luke has him born during the census of Quirinius. That census took place in 6 AD. So between the death of Herod the Great, the census of Quirinius, that's a 10-year period. And there's no way that Jesus could have been born before 4 BC and also in 6 AD, right? So my conclusion is not Q, therefore not P, modus tollens, right?
Yeah, I understand.
So,
So
and some
have you
people
looked,
say, well, there
Tyler,
was
at
another.
any
So,
Tyler,
can you say
have you
again?
looked at anybody that's been speaking about like the issues with that assumption that it was that particular census and everything? Because it's not directly spoken about, and there's actually issues with the way it's presented. So there's a guy on YouTube
Well,
called, he goes by Testify. If you look it up, he actually does a really good job of breaking down the issues with that particular contradiction.
it's like, oh, well, there was an earlier census. And here's the
Yeah, it's like
problem with
a
that.
separate census. Yeah, I agree.
Right, the census is a record, and if there's one thing that the Romans were good at, it was keeping records.
Okay,
And
wait, hold on, guys. I
and
want to stop this conversation because this is going into our religious beliefs. I'm trying to do science.
I
And so, like, I have another channel, Tyler. It's called Bread of Life. And I talk about all this kind of stuff on that channel. But, like, I'm
mhm.
trying to do a science thing here. And so I'm looking for scientific evidence for evolution. And it's interesting how like, and I know it's not just you doing this. I'm not picking on you, Tyler. It's like everyone who, like, whenever you try to get evidence for evolution, it's like they want to take you away from that topic and onto a variety of topics that don't correspond with evolution at all. So we're not here to, I'm not here to defend the Bible. I believe the Bible.
Right. Well,
I admit that it's a
right.
story I believe. So I'm not trying to
Right.
defend it scientifically, historically on this channel. I do think it can
Well,
be defended.
like I said, I don't know what, you believe, or if you believe in this. I'm just saying many creationists do. And the reason I bring it up is because this seems to be the main idea is that, no, you're wrong. The scientists are wrong. The evolution is because I'm right. And I'm right because the Bible's right. And the Bible's right because it's inerrant and so forth. And it's like, it seems like the only reason that you're going, there's pushback on evolution and so forth is because it conflicts with the biblical belief. And then people say, you bring up, okay, well, let's look at that. And then they say, well, that's, my religious beliefs. And we're not, and it's like, okay, you're telling me I'm wrong, and I'm wrong because, the Bible is correct, and now I'm not allowed to push back on that.
But that's actually not the reason why we're saying that evolution is wrong, though. I mean, it's kind of a straw man, to be honest. And I mean, you've been a very cordial and kind person the whole conversation, so I do like talking to you, Mr. Tyler. But just to be clear, we're actually pointing out that there are actual issues with evolution. It's just whether or not you accept those issues or not on at face value. So if mutator genome decay is actually a real process, which it is according to the NIH, then you have to contend with that as a mechanism within evolutionary history. And they have not been doing that on any of the papers. If you go and look at all the biologists and how they communicate the theory, you're going to see that they never once account for this. And that's a huge issue. It's called mutator genome decay or genetic entropy. And it's a real process. It's been documented for over a decade.
Okay, so the thing is, I mean, the reason nobody talks about it is because people that have looked through Stanford's data and have tried to study the things Stanford says is that he's not reaching adequate conclusions.
His name is Stanford, by the way, not Stanford. Yeah. But just to be clear, he is a PhD geneticist. He actually he's a creator of the gene gun, right? So he's the reason why we can actually produce mass crops and things like that.
Fantastic.
He knows what he's talking about. It's not just random information. He's actually produced this according to actual data that was presented to him, and not the other way around. It wasn't something he just threw out there and produced his own random data. He isn't fabricating bones and things like that to make it seem like paleontology has more than to offer than what it actually does.
So do you think Stanford is not imposing part of his worldview of the?
It wouldn't matter.
I'm asking that. Hold on. I'm asking that because you're saying Stanford has come up with this new scientific thing of genetic entropy, which I'm familiar with. I've seen what he's doing, and I'm pointing out. Look, part of the reason he's developing that is because part of his beliefs and part of the interpretation he's reading in Genesis post the flood is applying that interpretation to data. No, I am listening to you. I'm just pointing out what I've observed. Well, of course it's not his argument.
Then you don't know what you're talking about.
He's not talking about his presuppositions because he's trying to masquerade it as science.
Oh my goodness! Wait a second. So you're saying that because there are assumptions in the background, so if an atheist comes up with a theory, therefore he should be wrong just because he wants to assume a certain thing about reality? That's what you're saying.
I'm saying if Stanford wants to be taken seriously about the kinds of computations and back of the envelope. Hold on.
Okay, hold on. We're turning into dumpster fire here. At first, let me just say hi to Immutable. Good to see you. I'm really glad you came on.
Yeah, my point is that in any kind of conversation we have about science, right, like the process of evolution, the assumptions that occur are laid out very, very clearly in terms of what we expect, in terms of the kinds of mechanisms that we observe, that we speculate on, and we're able to make predictions. Some of them are good, some of them fall short. I did not see Stanford doing that when he's presenting these calculations, although I know exactly why he's doing it.
Can you quote any of his papers, like what they actually say, or do you just say this stuff?
So you're not disputing what I'm saying?
No, I'm absolutely disputing. I think that what you're saying is nonsense. I don't think you actually have read anything he's actually written about this stuff. And I don't think you
familiar
have
with what
anything
you're
about
talking about.
what the NIH has said about it. I don't think you know anything about what the NCBI has said about it. Nothing. I think you were just coming straight off the cuff, assuming that you know what's going on, and then just writing that off as if it's somehow a defeater of the argument.
I'm pointing out what I see when I have encountered his literature.
Sure. So what was the literature in question? What was the actual paper?
It's been several years. You tell me.
So you're telling me you actually did read this stuff, but you don't remember anything about it?
It's been several years. When's the last time you remember something off the cuff from 5 years ago?
If it was something pertinent like this that you're actually trying to debate about, I'm pretty sure you would remember it.
I'm not saying it's pertinent.
Look, guys, let me jump in and just say, I forget things all the time. I read a paper and then in a few days I might forget what it's about, what I read. So
all right, I'll be more charitable. I'll be more charitable.
Matt, you might have a phenomenal memory and memorize everything that's important to you. And in fact, it does seem with your level of knowledge here that you've read quite a lot on this, and you must have a good memory to have all that stored. So that's good. But it's okay that Immutable hasn't read that. But Immutable, do you have evidence for evolution that you would like to share? Who cares what Sanford did? Look, let's talk about what is the real evidence for evolution.
I mean, I guess I would need to clarify, like, what would you accept as evidence for evolution as you understand evolution?
Well, I would say I would like to see a mechanism that can produce new genes, not just move genes around, but actually produce genes. And so
I mean, I don't want to cut you off to kind of
no, that's fine. I'll just leave it at that. Go ahead. That's enough for me to say.
I think part of the issue here is that it may seem intuitive to a person who, like, if you're familiar with, I guess, like maybe Mendelian genetics.
Yes.
So part of the thing is that, like, I'm guessing the thing you're referring to is, like, if there's like point mutations or if there's gene duplications, that's not real information, that's not new novel information. Is that kind of the impression you're?
Well, in a sense, it could be, but I mean, I can concede that that's new if it's a duplication with a mutation. But what I don't see is happening is a process by which a gene that contributes to a new system, a new protein, something like that, it can originate by coordinated mutations.
So, I mean, I think the issue here is coordinated, right? Like when we talk about something like natural selection, okay, I would need an argument for why they need to be coordinated.
Well, that's actually what's going on with enzymes in general. When it comes down to it, your genes are being expressed in the form of enzymatic processes. You know that, right? And these enzymes appear to be doing things that are very sequential and very systematic. So it can't just be random the way you're assuming it, right? Ultimately,
you're talking about at this point the catalysis and the mechanisms by which enzymes operate. I'm talking about I'm talking about accumulation mutation within a particular, like within a protein of interest. And the argument that's being presented by Rebecca is that these mutations need to be coordinated in some sense. Well, I'm trying to understand why that's the case.
The reason I'm saying that's the case, Immutable, is because it takes thousands of nucleotides perfectly coded to code for a single protein. Okay, a single protein. Now, how did thousands of nucleotides just so happen to get together in just the right way to build a fantastic machine capable of accomplishing one of the many functions that need to happen for a cell?
Yeah, I mean, at this point, like the kind of question you're rearing at is more origin of life based. I mean,
no, because I'll give you the first cell. No, it's not. It's not, because I'll give you the first cell. So you guys say it all came from a single common ancestor, right? So I'm giving you a cell. Now, how do you get from a cell to all of the gene, like whatever minimum genes, and so far from what we can see, the minimum thing that we could get if we took the a simple organism and knocked out as many genes as we could. We could barely keep it alive with 480 genes. So where do you get the rest of the genes? If you started with a cell with 480 genes, how do you, and that's just a single cell, whatever, bacteria, then bacterium, how do you get to the diversity of life that we see? How do you get the rest of the genes that need to exist, that'll make your eyes, that'll make wings, that'll make all the features that we see in life?
Yeah, obviously I'm not going to be able to go through all of it. And there's different proposals one can give. Like one of the reasons, and even though I'm not going to talk about it that much, like one of the reasons we talk about, like, say, taxonomy and different kinds of like microfossils or fossils that are present in the fossil record is that on the one hand, like the question, for instance, about eyesight, given cells, there's various means in which proteins that are known are able to effectively respond to photons of light that have a given frequency, and they're able to tell when that kind of frequency of light is present and when it's not. And at the very, like, if you compare that to a pinhole camera, it's a very binary process, but it's able to give the cell information about, is there light, is there not. So it's at least directionality. And there's different processes. The fact that there's
information processing should be an issue for your theory. You understand that, right?
No, it wouldn't be.
Why not?
Because in this case, what information is being processed is conveyed by the conformation of the protein, and if there's some fluorescent product, etc.
You understand that information requires a specific language to be interpreted and then expressed. You understand that, right? It's what translation and transcription is all about.
That's one sense of the word information. What I'm talking about what I'm talking about information is I'm in this particular case for a cell developing some photosensitive pigment is, and this is one definition of information that can be given and that's useful, is that information is a process which allows something to perform better than random.
Well, who is making the judgment for what's better? Don't you understand what I'm saying to you? There's an information issue.
There's different ways. So, at this point, you're getting into mathematical modeling, right?
I'm actually talking about meta logic and meta ethics, that sort of thing. But regardless of that, the whole point I'm making is that you require a language to be existent before this is even a possibility. That's actually being computed and processed. You understand? It's just like syntactical values.
Yeah, what I'm saying is that if you're interested in the quantitative aspect of this, you can bake up easy mathematical models to compare. Like, okay, if you have cells that have photosensitive reception in some basic sense, what population should you expect to survive versus not in a scenario where, where cells are able to find food better if they're able to detect the presence of light. You can do these kinds of things, and there's no notion of a language you need outside of being able to mathematically cook up something.
What do you think?
Actually do quantitative. Yeah, the thing is, what we're talking about here versus what you're usually appealing to with semantics and genomics are usually regarded as slightly different in terms of like how they're used as notions.
No, that's actually begging the question. I disagree with that. And the reason why it's an issue is because you're trying to deviate from the point at hand, which is that there is information that's being processed in the systematics regarding how these things were expressed in the genome, right? And you just don't like the fact that that's the case. So you will try to rename it something else. It's not the same. It really, it can't be the same as that, but it has to be. That's the issue, is that in order for you to even have a process, there has to be a language to do so. You understand what I'm saying to you?
The process is literally chemistry. Like, if a cell can detect light or not,
we're talking about algorithmic processes. We're not talking about the chemistry. You can notate it using mathematics, but
the thing I'm talking about,
it's just a translation of what's taking place in the other language. You know what I'm saying to you? If you have a translator and they're translating what's taking place to another person that speaks that language, you see what I'm saying? It's the same thing. It's still information being processed the same way, but within a different language.
I would differ. I think.
How is that different? You're just begging the question.
No. When you talk about something, I mean, molecular processes are complicated. I'm not going to go into a lot of that. I mean, you could talk for hours about certain niches of molecular processes, but molecular processes, by and large, are deterministic. Not always, but by and large, they're deterministic. If a photon hits a photoreceptor, it causes a conformation to change from state A to state B in a particular way, and it gives an expected product. When we're talking about something like, the semantics of the genetic code or something like that, it's not at all deterministic, and it's not at all very, very clear. I mean, it makes sense in a very analogous way to a kind of code, but there's issues you can have of, for instance, a codon being free to be accessed by a transcribing element, but not being transcribed for some particular reason, right?
Yeah,
it's not quite as deterministic it's not quite as deterministic as a process as what I'm talking about with chemistry.
I'm not saying that everything has to be determined, though. You're asserting the determination portion. And by the way, that would just point to the fact that there is, again, algorithmic processes that are instantiated. Algorithms in general are always a mental information-based process. So there's no way to deviate from that point that I'm trying to make there.
Yeah, my point is I create a distinction between the kind of information that lets a cell determine if there's light present or not versus more complicated layers of genomic information. They're related in some way, but I take them as slightly different, as more nuanced concepts than that.
But where are you bridging the gap between natural processes and non-natural processes is what I'm trying to clarify.
I haven't mentioned the word non-natural processes here.
Well, okay, let's put it this way, like mental processes, where are you bridging that gap? Because there's got to be something there.
I mean, my understanding is Rebecca is asking for how certain information arises. One example she gave is that of the ability to detect of eyes, information, of which the first is to be able to detect light.
Right. She's recognizing that Shannon information does not bring about what we're talking about, is what she's pointing out. So you have to go above and beyond to something called
I don't think that's what she's
information.
About.
Well, I didn't bring that up, but that is an issue. That is an issue because every code that we see, we know a human being has created it. And so we know there's intelligence there. We know that it has been designed. And yet then we take DNA and people want to say that came about by natural processes. But that is an anti-scientific belief because
I don't think it is.
So, okay. So then if you find can you give me an example, like without appealing to something, that you guys assume is evolutionary?
You can't beg the question is what I'm saying.
I mean,
okay,
the question is to assume the conclusion in the answer to a proposition, which I'm not doing. I mean, I may be other logical because that's the definition of begging the question.
I'm saying, but how do you know you're not assuming it? Because every time I've heard you say something about this, you're already assuming that evolution's taking place, that this is how it works.
No, I'm not assuming that yet. I'm just saying Rebecca's asking for a process of how can X occur, and I'm trying to give an explanation for why.
Okay, let him continue. Matt, our complaint is registered. Okay, our complaint about information is registered. Now, Immutable may not agree with it, but it's registered. So let's let him continue on the process of how do we get the genes and everything that needs to go into vision, for example. Okay.
Sure. So, I mean, I think the point he made is good. I mean, because one of the points I agree with is, look, you need to be able to test, you need to be able to discern if a mutation is useful or not. And ultimately what helps determine that is the environment of the organism finds itself in. I mean, Rebecca, if you're willing to accept that, for instance, you can get that photoreceptive processes can develop and occur in a cell that doesn't have that capability, then having other things occur.
I haven't.
Do you not accept that?
No, I do not accept that. What I see at my scientific observations are that what we observe in organisms comes from what is already present in the genomes. Now, there's a lot of potential within our cells and to adapt to the environment, but we move genes around. Okay, we might be able to get genes to move from one thing to another by horizontal gene transfer, but somewhere you've got to get all of this genetic information. And so I'm asking how genetic information, and then not just the information, but you have to get that to manifest in like machines, systems, integrated systems that will work together in harmony. Now, I'm not seeing how that happens by single nucleotide changes over however long. You can have as long as you want to do it. That's not what we observe happening in nature.
I mean, I don't know if this is a thing you're trying to just be clear. Are we talking about the origin of life? I mean, this is a kind of implant. Okay.
I'm giving you this. I'm giving you the first.
We already
cell.
said the cell exists, but even though that is actually part of an issue.
But so in that case, I mean, are you not granting a certain level of like basal metabolic and genetic regulation and certain things like that? Like already
I'm not granting you that in a single cell, but
okay.
but getting from a single cell to the things that we observe is going to take a major feat. So I'm asking, how do you accomplish that feat? How do you get all of these systems to form? And so I'm granting you the cell, not because you deserve it, because evolution and abiogenesis should not be separated. This is the theory of natural origins. But because people can't take the heat, that's why we've allowed this separation of abiogenesis and evolution. I shouldn't grant you the first cell. I'll grant you the first cell. Now, use the first cell. Get me to where we're going with the first cell.
Yeah, I mean, by and large, that's the easier component. I mean,
okay, so you're saying the first cell had photoreceptor. You're saying the first cell could sense light. Is that what you're telling me?
What I'm saying, okay. The first life, we don't know what it is. There's different candidates for what kind of life is what people refer to in the literature as the last common ancestor of all life. It could be photoreceptive. It could be one that was able to detect light. It could not be. There's different arguments people will give. So to the point you made, coincidentally, it turns out that the class of molecule that is responsible for detecting light and its presence is very similar structurally to molecules that already deal with things like transporting metal ions around in the cell. If I recall right, these are classes of molecules called ferrocenes. These kinds of molecules are ubiquitous and made all the time in the cell. And so what you have basically is just a different utilization of a certain kind of biomolecule for a process. So it's not that difficult in this case to be able to have a photoreceptor develop for a kind of cell that's exposed to light decently often.
Okay, and then how do you get from that to the variety of eyeballs that we see today?
Right. So if you have a cell that's able to detect light, the next thing you're able to do is that as mutations accumulate, right, certain processes that develop in a cell, or certain things that are present in a cell may develop in such a way that rather than being able to detect light is great, but it doesn't tell you about what direction that light is coming from. So different parts of a cell that are used for structural purposes may be able to develop in such a way where now, rather than detecting light at all possible angles, the light that's being detected has to enter through a certain angle. And so this is the development of basically a very blurry camera of not just a cell being able to detect light, but also being able to detect where that light is possibly coming from. And
okay, but do you realize that right now what you're doing is you're telling me a story?
It's all the engineering language too.
So I need more than a story, because I don't believe this story. So,
okay, so I
I
mean,
need
you
like
can,
actual evidence that this story is true. Why should I believe the story that you're telling?
right. So what I would encourage you to do in this case, I mean, I'm not a biologist, for one thing, and this is stuff I learned like back 8 years ago in a college course. What I would encourage, so what I've done is I've given you propositions about different things that can occur regarding certain kinds of photoreceptive cells. And the prediction you can make is that, hey, well, if this is true, maybe in nature somewhere, different kinds of cells or different kinds of animals have these different kinds of eyes that we're talking about, right? And I mean, to the best of my knowledge, I mean, if you look up, if you type in a search term like the evolution of the eye or something like that, or evolutionary mechanisms for the evolution of the eye, you should be able to reasonably quickly be pointed to a general kind of discussion like the one we're having now about, at a heuristic level, how this process could occur. And also, it probably should give you, at the very least, some examples of organisms which have differing levels of complexity of eye. So organisms that are just able to detect light, organisms that are able to, where effectively you're developing a very weak lens of being able to detect light from a certain angle. And there's tons of research that have been done in regards to how exactly like photoreception in the eye works, and also how certain processes get refined when it comes to seeing things.
Well, I have looked this up actually, and what I get is a lot of evolutionary storytelling. And
well, so to go to the point of the eye, do you find organisms that have differing complexity in eyes and what those eyes are able to do?
It doesn't
absolutely.
matter. What does that
Right.
have to do with anything?
Well, so this puts what I'm saying beyond the level of just hypothesis, right?
No, it doesn't.
Well, it doesn't.
No. We can dispute, hold on. We can dispute different mechanisms that occur for it. There's different ways you can test if certain mutations are responsible for it. But the thing you've said is that, oh, I have looked, and yes, there are various stages of development that correspond. There are different organisms that have these different kinds of eyes in the
First
kinds
of all,
of ways that I said they should.
sir, this assumes that all of them are in sequence from organism to organism.
No. So
Yes, it
no, I
does.
didn't
It
say that.
automatically assumes that. This
I
is what I'm trying to point out to you. Your assumptions automatically assume evolution from the start.
No,
Don't you see the issue with that?
no, I didn't.
Yes, you do. Every time you've made a statement here, it's always under the assumption that it's going to evolve into something greater.
No, I
And
haven't.
mind you, we're talking about more beneficial over time. That it's another organism coming from another organism that actually has even better genetic sequences.
Unfortunately, we haven't reached that stage in the conversation. The only thing I've talked about so far is the propensity for mutations to occur, and if said mutations occur in a special kind of way, what should we expect certain mutations to do, and if those things are accurate, if we can observe them in nature. And the answer Rebecca gave is yes. You can dispute
she
to a certain
admitted that
degree.
there are different type of eyeballs, is what she admitted to. She didn't say that that was somehow evidence for evolution. You're trying to point it as evidence for evolution and try to put them into some kind of hierarchy that says they're supposed to come from this, that must come from this, that must come from this.
No, so
that's already assumed in what you're talking about.
No. The point Rebecca is asking about is how new information can arise. What you can do now that you have different candidates for eyeballs is you can do genomics now. You can test what are certain genetic elements that are common in all parts of organisms that have this general kind of object called eyes.
Immutable, but that assumes, right? No. You're still assuming that there's an evolutionary connection.
No. I'm not assuming that. I'm saying what we've agreed to so far is that you can have cells that can detect light and those that don't, right? And then what we've been saying is there are different other kinds of mutations that can occur in such a way that improve an organism's capability to detect light in various kinds of ways. I haven't said yet.
Here's the thing I wanted to point out, sir.
Hold on. I haven't mentioned anything yet deliberately about how these organisms need to be related to each other. All I'm talking about so far is what we have and what different things can and what kinds of mutations might be able to lead to it.
So she started off with a basic cell. She didn't like say that they had the ability to detect light or anything like that. You just kind of came in assuming that from the start. That's one thing to begin with. So that's one issue. The other thing here is that she's trying to point out to you that we are trying to start from the very beginning. Once a cell has finally come to existence, right? That's what she's talking about. She's not talking about maybe
Right.
in your theory, this would have been several, probably even a billion years before what you're even assuming right now with the light sensitive cells and things like that. So I'm like, that's just assuming that your theory is even. That's what I'm saying. You're starting off under the assumption that this is already taking place for many generations.
So basic logic 101 for you.
No, this is
if you're going to entertain the idea that certain processes can occur, you need to at least grant me the fact that A, life occurs, which Rebecca did agree to, and 2, that at least such things are plausible, which so far Rebecca has agreed to.
Wait, I agreed that what was plausible?
She was asking for a mechanism to get to those things.
Yeah.
She didn't ask you for a assumption that those things occurred. That's what you're doing. Is you're assuming
the only assumption is that mutations occur.
No, this is what you've been doing. You've been pointing to things that do exist and then saying that those things exist, so this had to happen because I assume this has already occurred.
I have never used the phrase "had to happen." You can watch this conversation back as many times as you want.
Your logic
and I will never have used
automatically assuming that.
Look, I appreciate you being here and that you are having an emotional response to what I'm saying, and I'm sorry that that's occurring. But if you're not able to represent my point correctly, I don't really have a point to try and engage with it, because me and you are making different points, and you're throwing in statements that I haven't made.
No, I'm pointing out your underlying assumptions, and that's what you should be doing.
I haven't made the assumptions. I'm very clear about the assumptions that I'm making.
No, we have to point out to you what your assumptions are, apparently. That's the difficulty here.
The assumption I'm making is that mutations are possible and that said mutations have an effect. That's all.
Okay, we can agree that mutations have an effect.
Right. So now the thing you can do is in different
organisms. Go beyond?
Okay. Yeah. So now the thing you can do is in organisms where you have these different kinds of eyes, you can do genomic sequencing on various regions, on the various elements that constitute that eye, right? So you are able to tell, okay, so here's the kind of gene that's responsible for the pigment. Oh, and okay, so in the kinds of cells that are able to have some ability to detect the directionality of light when light is coming in, oh, so it's this family or single cluster of genes that's occurring. Oh, okay. And we see this gene, and we also see this, so we see the gene that has the pigment, and we also have a gene it's a gene or family of genes that's responsible for refining that into a more precise process of detecting light. And you can very quickly be able to tell how these different things are similar and or not similar to each other. And it turns out to be the case that when you do this, these family of genes that are responsible for eyes form a nested hierarchy.
Okay, and so now how does them being similar show me the mechanism by which they can come about?
So you're asking, so one of the questions you're asking is about how do you get new information? So one of the things you mentioned earlier was about if you have a certain, so basic genetics, once you have DNA, you have a certain sequence of nucleotides, a lot of stuff happens, it eventually gets transcribed into a protein. That protein folds, it has some function. Now, one of the different ways in which you can account for different new proteins arising, and there's textbooks and there's different means by which it can occur. I'll give you one common pathway that these things can occur. So, and this is observed quite frequently. So when an organism, are you familiar with the term genome duplication?
Yes.
Okay. So basically, the reason this is important is certain genetic accidents, like genome duplication, can occur. And if they occur within an organism, it can actually be a tremendous source for new variation to arise for the following reason. You have two copies of the genome that have now been put together in a single genome, right? Because the genome of an organism is doubled. From an evolutionary point of view, what is important is that the genes that are essential for an organism to survive stay. And now, since you have twice as many, since you effectively have a second copy of that genome that has basically these spare genes, right? Because they're already present in the original genome, they're free to undergo mutation to no cost to the organism at all, right? So some of these mutations may destroy the gene. Some elements of one gene might get transferred into the other gene, and this can cause new families of proteins to arise. You can have different other things occur where even if the protein stays pretty much the same, where things that regulate genomic activity are changed. So how something gets expressed changes previous to how it was before the duplication occurred. And
okay, now immutable, you're giving me gene duplication, and I agree that gene duplication occurs. Okay, but beyond that, what are you telling me that is actually scientific evidence rather than storytelling?
Right. So the scientific evidence is when we were talking about genomic sequencing and stuff like this. Right.
Well, I don't accept that as evidence, because I also understand that organisms can have common genes. It doesn't mean that they had a common ancestor.
Okay, so in that case, I just view that. I mean, to what I understand of your view, Rebecca, I just don't view that as parsimonious, right? So the one issue that I see with creationists and part of the model, right, is if there's something that doesn't seem to make sense, you can say, oh, well, God chose to do it in a different kind of way, right? And I'm not saying that's wrong. I'm not saying you shouldn't be able to do that, right? Part of the thing with evolution, right, and the story that evolution tells, right, is that if certain things are related, you do expect for these similarities to show up, and not just these similarities on a generic level, but certain kinds of similarities being organized in the kinds of way that the theory is predicting they should occur. Right?
I accept that as parsimonious with your story, but now Grayson would like to jump in.
Well, and so where I'm leading this, and I don't know, like I'll take turns with Grayson, is that part of the interesting aspect of this is that it's not just the functional aspects of the genome that are interesting, right? There are, you can find different genes where if you look up certain parts of the gene that are like the gene that's similar, right, but the mutation hasn't rendered it useless. It does something in it does something in a previous organism that the new organism no longer has any function for, right? There's effectively, certain genetic litter in some organisms that shouldn't really, or doesn't really seem to have any reason a priori to be there, right? In a certain model of creationism. But if evolution does occur, right, and certain organisms are related through certain aspects of genetic similarity, if that gene comes and goes, that's fine. We understood how it got there. We understand how it got disabled.
Okay, thank you. Grayson?
Yeah, hey, Rebecca, long time no see. As long as we're going over evidence for evolution, I do think that the patterns of genetic similarity by it of themselves are sufficient, even if we didn't have any fossil record. But specifically, I would point to the congruence between the fossil record and the patterns we see in, nested patterns of genetic similarities. Specifically, I also think that your conversation last week with Dr. Dan I thought that he presented some pretty foolproof evidence for evolution in the patterns of genetic similarity that we see that don't seem to be able to be produced by any model that involves the created kinds, right? I mean, he was showing that the patterns that we see when looking at the genetic variability in different organisms, not just on the gene level, but in like the non-coding DNAs and the patterns of their actual mutations, those patterns are not replicable with a created kinds model. If the pattern of created nested hierarchy that we see was created at the start in these kinds that are, phylogenetically an orchard instead of going back to a single root, then the patterns of mutation that we see are incongruent with that model, as in it rules it out based on the scientific evidence.
Well, I haven't been shown that yet. Actually, I know that you think Dr. Dan presented good evidence for that, and maybe Dr. Dan does have something that he can show me. So I actually sent a private message to Dr. Dan to ask him to clarify and actually demonstrate what he's talking about, because, and I pointed out the flaw with what I thought was the flaw in the demonstration that he did. Okay, so, and he's here and he thanks you for talking about bringing up what he brought up.
Nice.
So I'm not ruling that out yet, but for me right now, I have not seen and the main problem that I'm having with this is that you guys are assuming that if something doesn't have a common history, it would have no genes in common. So
right. So this is not on the gene level. I want to clarify.
Okay.
Like, we're specifically talking about like mutations to the entire genome, not just between genes or whatever. But if we're looking at like the whole genome and we're looking at, oh well, here's a mutation in this region, right? And you could see that certain species share certain mutations in the same region, and that either has to be a coincidence or they're both inheriting those mutations in that specific location on the genome from a common ancestor. And so you can test those two models. And statistically, the odds of just all these patterns of mutations that we see in different organisms' genomes just coincidentally being the same mutations, if they don't share a common ancestor, is like less probable than you picking one specific atom in the entire universe. I mean, it's just statistically impossible.
Okay, I read the paper that said that. Okay, and it's very problematic. And Matt, I'll let you jump in just 1 second. Just let me say the last thing that I want to say about that, which is, Grayson, everything that you're saying is on the assumption. See, if you have common ancestry, then you had to get everything through mutation, right? In some, or whatever, gene duplication, whatever, but you have to, all the changes that you're seeing have to be mutations. But in my model, there can be differences that are not mutations, right? And also not recombination. Not recombination, like that were original to the organism. So you're assuming your assumed starting point is what's causing you to see these coincidental changes.
Right, but we both agree that mutations do happen, right?
Yes, absolutely.
Okay, so then you have the issue of figuring out which differences are due to mutations and which differences are due to some sort of created design differences inherent to these kinds of organisms. And it seems like all of the differences that replicate that pattern of nested hierarchy, you then cannot label as mutations, because then you have to give up your position. But if it just seems kind of like a no true Scotsman fallacy, where if you find a difference, you rule it out as being a mutation because of your worldview assumptions.
Well, and let me steelman Grayson's position here, if I understand it right. I think part of what Grayson is saying is that when you lay out the genetics of an organism, for instance, and you're trying to say, like, in a kind of created kinds model versus an evolutionary model, what is the thing that's telling you of something, right? That if the similarities are occurring are the result of God choosing to do whatever he chose to do versus mutation, right? And the problematic aspect of that is going to be, in certain organisms, you may see certain similarities, right, that make sense of, like, oh, well, of course, of course, cheetahs and let's say leopards have similar mutations in things that correspond to their eye because they both belong in the cat kind, right? But certain other things that that you may not accept as being related to each other, like the eye of a cheetah and the eye of an octopus, even if they operate very similarly, right? You would say, oh, those mutations aren't necessarily that. That's just what God chose to create, separate creation, right? And it seems like the basis by which you're choosing to do that is arbitrary, right?
I'm not saying that I can scientifically determine that, right?
Well,
right. I think Matt wants to jump in. So let's let Matt respond to all of this, if that's okay.
Yeah, I just wanted to speak a little bit to what Grayson said. And I mean, he had a very thoughtful response. I appreciate it. But I just wanted to say that, so I don't know if you saw the video with Dr. James Tour and Rob Stadler when they went over that information. And Rob Stadler actually laid out a very thorough refutation of what was taking place there and explained about how they had many assumptions from the start of the paper that are already in depth explained in the paper, but you kind of had to go do some digging to verify it. And it obviously already assumes a certain model of creation before they ever try to, attack it, of course. And then it gives them that p-value that was extremely, high that you were talking about, where it's like, you couldn't even select one atom in the entire universe, especially compared to this other thing. I understand that's a pretty crazy p-value. That's insane, right? But that's assuming that they wasn't using something like the dependency graph model. That's what Rob Stadler had brought up, which actually apparently was highly favored over the tree of life model, right? So if you had gone and verified that, you would see what was going on. And I'm not sure if you had actually got to hear that or not, but so the dependency graph model actually is similar to like what software engineers use. Essentially, they will go and they will take these dependency graphs where they have multiple sets of code, and to perform a specific task, that code is already preset. It already has like a certain base, code set, right? And so the programmers will come in there, they'll take a portion of that, whatever is required for their job, and then they will modify it according to what they're trying to implement it in for that particular job that they're doing, right? And so those different things might get put back into the overall dependency graph, but you can see that that's where they were drawing from, was the original code, right? And it could be the same in that model when it comes to what God could have done there. Now, just to be clear, this doesn't prove creation or anything like that. I'm not saying that's the case, but just to be clear, it's just like, you can't rule it out. That's the biggest issue here. And it seems like it's all automatic a priori. They were assuming evolution without us at least giving the designer a chance to do what he's supposed to. And that's what every designer today is doing, just to be clear. So it's like, why would we assume that the designer would do anything different, if not better, than what we're doing right now, right? You see what I'm saying?
Yeah, so I've read the paper. I've seen Dr. Dan's simulation that he did that was similar to the idea in the paper, but I haven't seen Stadler's response. But from what you just conveyed about it, it doesn't sound like it's accurate because, I mean, the paper did not just assume evolution, neither did Dan's model. I mean, they're testing two different models, one of which is the created kinds model that doesn't assume evolution, right? What it assumes is that God created the nested hierarchy pattern that we see today in genetics in that same nested hierarchy at creation. And you had these separate created kinds that were fitting into a genetic pattern of nested hierarchies. And then the only thing that goes from there is that the species mutate. And all of us agree that species mutate. So we're just saying, if you have a starting point that is the creationist starting point, or however you want to do separate created kinds, you can literally input it yourself into Dr. Dan's model and see what the results you get. As long as you have mutations, that's all that is required to rule out creationism. I'll look at Stadler's video or response to it. Maybe
I would recommend. I'm sorry, not trying to cut you off. I just want to say I would recommend just taking time to listen to what he actually said, because I don't know if I represented it properly, but what I was pointing out is not that they're assuming evolution to start. What's happening is they are mischaracterization. They have a mischaracterization of what the creationist model is. They're not presenting the most accurate representation of how a designer would do something. They are assuming specific ways, and then they're strawmanning that position and saying that therefore that can't be true, and then they're applying it to every single design model that could possibly exist. You see what I'm saying? That's the issue. So that's why I would say, look, I understand you can have your model, and in some sense it could work. But I also want to contend with what Dr. Dan has not done so far, He has not been presenting any models that actually take into account mutator genome decay, right? And that's a huge issue because even the NIH is recognizing that. So it's been in their literature for over 10 years now. They've been talking about it all over the place, but people don't bring it up because it's a pretty detrimental thing to the evolutionary theory. Ultimately, what's taking place there is they have shown how every single genome, if it's not identical to its parent genome, ultimately has a net loss of genetic information per generation, right? And if that's the case, then that actually throws a huge monkey wrench into your whole theory. Now, they don't talk about that, and obviously you can see it's not his models, right? You're not seeing the genetic mutation with decay over time. You're actually seeing the assumption that there's some kind of producing of new genetic information that's going to be functional in the future, and that's an issue. I'm not saying that there can't be functional information. The issue is that we're always assuming it without taking into account the issues with that, right? I want to say that that's what's going on here.
Oh, well, it looks like we have the author of the
big
paper.
dog's coming in. The big dog's.
Hey, the man himself. The man, the myth, the legend.
Chat, and I had to jump in because I couldn't type fast enough to say all the things that I needed to say in response to what I'm hearing. So how is everybody doing?
Good.
I'm great. That's fun.
Okay, so can I just say real fast, two things from the last like 5 minutes of this that I was explaining in chat, but like I said, I can't go fast enough to keep up with the conversation live. So first, that paper, Rebecca, the paper that we're talking about, the simulation with the super low p-values.
Yeah.
They didn't pick a starting state. There was no assumptions about starting state at all. And if you dive into the methods of that paper, what they did is they said we have several different models that we're evaluating, right? There was species, separate ancestry, family, separate ancestry, and then universal common ancestry were the big ones. And what they did is they said, okay, we have what exists. We can directly document that. Given the processes, what's the likelihood? And they used two different models. There was a parsimony and a likelihood, and those have like very specific definitions, but it basically is what it sounds like. Parsimony is fewest changes, and then likelihood takes into account things like the asymmetrical, mutation rates, that kind of thing. Like C to T is more likely than T to C. And so they said, given what we directly observe in terms of existing genome sequences and patterns of similarity, and what we know about how genomes change, then what is the likelihood and what are the most parsimonious processes or parsimonious histories that get us from origin to current state? And what they found was that common ancestry is very parsimonious, highly likely. Family and species, separate ancestry, non-parsimonious, highly unlikely, right? And again, there's no assumption about the starting state because the way those models work is they're iterative. So you keep trying different variants of like, the computer generates a lot of variation, and then it's almost like a natural selection kind of process where you generate a bunch of phylogenetic trees and kind of pick the best ones according to each model. And then each one will converge upon the best trees. So there was no assumptions built into that about the starting state. So I want to be clear about that.
I don't see how you can not build an assumption into the starting state, though.
The way you do that is I say this not in any malicious way, but if you're not doing graduate level phylogenetics, you wouldn't necessarily see that because you're not looking under the hood of how these different things work. But if you look at how the different ways of doing phylogenetics work, there's different methods of doing it. And the way they did it is looking at existing diversity and working backwards while being agnostic about what that initial state was.
Okay.
So, Dr. Jen, I have a question.
And then second thing that I was trying to type really fast was to Pigs Can Fly, talking about how I did not take into account things like genetic decay in my thing. I didn't have audio for just a couple seconds while I was joining StreamYard because it was muddled and overlapping, but Pigs Can Fly, is that a fair representation that I'm not considering like genetic decay in my simulations?
That's correct. Yeah. Yeah. So there's,
Okay.
as I mentioned before, the NIH
you-
has already spoken about this over
Yeah.
a decade
Are
now.
you familiar? Are you familiar with the paper that I co-authored last year with Dr. Zach Hancock?
No, actually, I'm not. If you want to send it to me, I can take a look at it.
We refuted genetic entropy, which is all the genetic decay stuff is genetic entropy. That's what it is. And we mathematically refuted that using empirical distributions of fitness effects, which is to say, using experimentally determined distributions of the fitness effects of mutations as they occur. We showed mathematically that genetic entropy is wrong. Like, that is a paper that was published in the Journal of Mathematical Biology last year. Dr. Zach Hancock was the primary author. I was the second author. If any creationist, you or anybody else, wants to say that genetic decay is a thing, it's a problem, it precludes evolution, whatever, you need to refute the math in that paper.
Okay, I'll certainly take a look at it if you wouldn't mind sending it to me. But I just want to clarify. So you're telling me that the NIH got it wrong whenever they were publishing that information over a decade ago, and they've still been talking about it, by the way, all the way up to this point.
Link the source, the NIH source, in the private chat so I can look at it. I think I know what it is.
I mean, you could simply type. You can literally go to Google Scholar,
link me, dude. Don't play
type
that game.
in mutator genome decay, and you will start seeing.
For the love of God, why don't you stick by your source and just link it?
I can't read your mind. If you want me to tell you if I agree with the source or not, tell me what the source is.
I just told you it was the NIH multiple times.
No, that's not good enough. I need title and authors.
To be fair, like you just asked
Dan.
Listen, no, I can't just leave and then come back. It will give me a hard time about that.
That's what I'm pointing out. You just demanded that Dan send you a link. You just demanded that Dan send you a link.
If you want me to tell you if I think your source is wrong, tell me who your source is. Give me a title and the authors, and I can look it up. But if you
you know what? We can do that on another stream too. If it's not something that Matt can get right now, then we can get it later.
I linked our paper in the private chat so everybody has access to it if you want to look it up.
Yeah, I'll definitely
and I can put that. I will put that in. Do you want me to put it in the public or no?
Absolutely, put it in the public chat. Absolutely, it's out there. It's
I assume that Pigs Can Fly was probably referring to Dr. Sanford's talk that he gave at the NIH.
No, that's not the only thing. I mean, he, of course, is one of the people that's talked about it, but there's many people.
That's just genetic entropy, though. Then you go back to the paper.
It's labeled mutator genome decay by the NIH. It has nothing to do with
mutator genome decay. Stop right there. That's a Lenski thing, and the problem there is you are saying that because the mutator lineage has higher mortality, that it's showing decay, but that's wrong, because what happens is that the mutation rate goes up in the mutator lineage because they lose their DNA error correcting mechanisms. So they have a higher inherent mutation rate. What happens is the average fitness of that population goes down, but the maximum fitness goes up. And you get this dynamic where you're constantly finding the most fit genotypes, but then because you have a higher mutation rate, you're also mutating away from those most fit genotypes. And this means that the most common genotypes are not the peaks that have the highest fitness, they're the peaks that have the closest to 1 or 2 nucleotide difference fitness. So it's this whole dynamic called survival of the flattest rather than survival of the fittest. It refers to the shape of the curve. It's a flatter curve rather than a steep curve. And that's a well understood evolutionary dynamic under high mutation load. Important thing with that is that does not mean those populations are decaying or going extinct. It just means they have a different equilibrium point. And again, I'll emphasize the maximum fitness is going up, even as the average fitness is going down in those populations. So that's not an example of genome decay. What other creationist talking point would you like to throw at me while I'm here?
Wait, I want to go back. I don't want to miss this opportunity because I still want to go back to the separate ancestry in this paper. Okay, because I know you're telling me the reason I can't understand is because I don't do graduate level phylogenetics. Okay, but that's not going to suffice. Okay, like I can be made to understand what the mechanism is that you guys are choosing your.
Okay.
And you're saying, wait, hold on. you're saying it's agnostic, but
Yeah.
to me, I don't
Yes.
see how you can be agnostic about your starting point. If you're calculating 4 separate ancestry, you have to be assuming something at the beginning. Now, what I think, what I could tell, from what I could tell, you can correct me, from what I can tell about the paper, it looks like they're assuming separate ancestry by natural processes. So am I wrong about that?
The process is, so what they're, the only assumption built into that paper is that the processes that occur from start to present are the processes that we observe today and are operating as we understand them today.
Okay, so do they do like, then I feel like you would need, if you were really going to do that scientifically, you would have to like have many starting points that you start with. Like, meaning like, okay, if we start with separate ancestry, we could start where they have these genes in common. So you did, okay, so how many different
what they do,
ancestry methods?
There is no single number for that, because the way these models work, the way they describe it in the paper, is they say, here's the existing variation and here are the processes. So given those two things that we're locking in place because we can directly observe them, right? We can sequence genomes and we can observe how mutations work, right? So we're going to lock that stuff in place, and we're going to be agnostic about all the other stuff, how it started, what it looked like when it started. We're not going to take a position on that. But for the different models, we're going to say for common ancestry, we're going to say all these things have to go back to a common ancestor. We don't know what that looks like, but given the starting state and the processes, how likely is it that that's possible in the, given our constraints about like mutation rates and the relative mutation rates and deletions and insert, right? Where all the processes we know. For family separate ancestry, they're taking this group and this group and this group and this group and saying, okay, each one of those has to go back to a starting place. And then that overall pattern that we see has to match the existing pattern that we directly document. And for species separate ancestry, they're doing the same thing, except they're starting with each individual species as it exists and saying, okay, given these processes, you have to get there, but you're starting from independently, right? You're starting in a different lineage and you have to work your way down. Now, what those independent starting points look like, the paper is agnostic about it, because you're working backwards from the present, and the models for each experimental run are going to converge on the best possible starting state for each of those models, right? So like the p-values for the separate ancestry model are the best possible p-values that you could possibly get, assuming like the optimal starting states for those models.
But okay, and I mean, I really hope you'll like just, if you, okay, I look, it is still assuming that those genes, common genes had to, or common genetics had to have a common history.
Nope. It explicitly.
I don't see how
it explicitly says they
I feel
don't.
like we are either totally talking past each other, because like I'm hearing you say this, I'm understanding your explanation, but your explanation still is not addressing the problem.
It's just, let me try explaining it a different way. So what the three tests, so you've got common ancestry, family, and species, right? You've got your three options. And what each one of those kind of iterations of the model says is, what's the likelihood that if we start at the same point, we get the current level of diversity? What's the likelihood that if each species starts independently, we get the current level of diversity? What's the likelihood that if each species starts separately, we get the current level of diversity? And in order to answer that, we're saying, well, we know that mutations occur at this rate with this distribution of likelihood and blah, 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 right? And because, again, once you start, you're just operating with whatever observed processes we have, mutation, recombination, et cetera. So the model is not positing, and this is where it differs from mine, right? It doesn't say, here's what the starting state is. It doesn't do that. It just says, if we assume separate starting states, what's the most likely scenario that gets us to the current state?
And what do the starting states look like for each organism?
They don't. There's no other way for me to say it except to say there are no fixed starting states. There's no defined starting states in those models.
So you don't have anything to compare it to. If you don't have a starting state, you don't know what you should expect.
Can I try this
Grayson,
one?
if you want to try, go for it.
Yeah.
Okay. Like, so you have the common ancestry model. We got that one. The things that they're comparing it to are if each species did not share a common ancestor. So what does any possible starting state look like that gets you the results that we have today for each species if no species on Earth shared a common ancestor with any other species? And then they try another one with the family level as saying, what about if each family did not share a common ancestor with any other family? And they're not assuming anything about what the starting states look like. They're just saying, well, given all the different possibilities for what these starting states could look like if they did not share that ancestry, then what is the likelihood of us getting the results that we have today?
And how many starting state potentials did they calculate? What
That's
I
just-
saw was 2.
I want to be clear about this. They didn't calculate starting state potentials. They didn't do that. The way these models work,
And I'm saying that's the problem.
Wait.
Okay.
No, the way these models work is it is an iterative process where the computer generates a certain number of outcomes, right? And then it evaluates the validity of those outcomes according to the parameters that it was given, which for this model are the relative mutation rates, the frequency of other genetic changes like duplications and such, and the existing pattern of similarity, right? Existing sequences, right? And what it does, it says, okay, we're generating all these potential pathways from A to B. How well does each of those match the parameters I've given? Are they highly likely or are they highly unlikely? And then what it does is it takes the most and you can do this there's different ways that some versions of this, you take like the 4 best out of 1,000. Some are a little more permissive and you take the top 10%, whatever it is, right? You take all your iterations and you say, okay, we tried this 1,000 times, right? Let's take the best 10% of those 1,000 trials. And we're going to start with those where we get the best match, right? The highest likelihood. And we're going to mess with those now by a certain level of change. And we're going to evaluate those 1,000 possibilities and take the best 10%. And we're going to then vary them again and take the best 10%. We're going to do that 1,000 times until we converge on a best answer to the question, right? That's how it works. So you're not telling the program, here's what the starting state is. You're letting it find the optimal starting state for each hypothesis.
Yeah, effectively what it's doing is like, I imagine like the number of possibilities that accumulate grows it grows exponentially. And basically what it's doing is it's saying, okay, based upon the certain set of constraints that the code is running against, right? Here's the best after a certain number of iterations. And from the best in iterating this process a certain number of times, what is the best what is the best possible thing you can get out of this, right? And in a way, I mean, it might even be overly generous because what you're doing is you're saying the best of these, right? The best of these stay, the rest of these you're no longer, right, worried about, some of these computations and back propagation, like with the program of computing, like what could have given rise to this. Right.
Okay, now let me tell you what I'm hearing that this experiment is capable of. It's capable of showing you whether common ancestry is a possible explanation.
Mm-hmm.
Okay. And it's showing that common ancestry is parsimonious with what we see in genetics at that level, right? So that may be what it's showing.
It's showing that it's both parsimonious and high likelihood, which are two different
things. That's where I'm having the issue. And Dr. Dan, let me try to explain it in another way, because I thought about your demonstration. So maybe this will help you understand where I'm coming from. Let's suppose we did that demonstration again, where you did the separate ancestry versus the common ancestry. And you offered to me, hey, we can change the parameters if you want. Okay. Now,
Which I did.
I know you did. Now, so let's suppose we're doing that again. Now, and we don't actually have to go through it I hope for you to understand the point that I'm having. What if the parameters that you used for common ancestry, we used that in the separate ancestry? Like, what if I chose that as the parameters for the separate ancestry?
I'm not sure what you mean, because the parameters for common ancestry were just that they share common ancestry, which is the
okay.
Right, that's the
Why
point, right?
do
Like
those parameters get to be the common ancestry? Why can't those parameters be the separate ancestry?
What do you mean when you say parameters? Do you mean starting points? Do you mean
The starting
changes
point.
after?
The starting point. I mean the starting point. The part
here's
that
why.
you
Here's why. Because in the separate ancestry model, you're limited in the number of variation that pre-exists in each lineage. That's why.
But are you? That's what I'm
Yes.
challenging.
Yes, because you have a maximum of
That's what I'm
4
challenging.
alleles per locus. Yes, you are limited by the number of individuals that get off the arc. Yes.
But you're not limited. Now you're limited within one type of organism, a kind.
Yes,
Okay, you're limited within a
yes,
kind.
Exactly.
Okay,
Yeah.
good. Matt's back. I'm not alone against 30 people.
No, hang on. Let's finish this point. You're limited within a kind. So keep going.
So, all right. So you're limited within a kind, but remember, your demonstration was not a comparison like of alleles within one kind. Your comparison was 3 different kinds.
Right.
Okay. So your comparison was 3 different kinds.
Right.
So why can't those different kinds have the same starting point that you used for common ancestry? This is what I'm trying to say. They may have common genes that don't share a common history.
Because you have to generate all of the within lineage variation after creation. That's the point. Is like there's like humans in the ABO blood group gene, we have over 1,000 variants of that gene. But at creation, there were 4 variants. So you have to generate all that variation. You know what I'm saying? So in the common ancestry model, some subset of that variation accumulates in the common ancestor. But in the separate ancestry model, all of that variation, the vast majority of which must be due to mutation, must accumulate in each lineage separately. That's why.
Yes, and like I said,
Rebecca, let me just ask a quick question. Do you disagree with that last sentence that I just said?
No.
Perfect. Then we're good.
Okay, but so I'm saying within a kind. So if you want to bring the blood group thing and do it where you actually account for mutation and recombination, and then it's still showing that we can't get those alleles, then I'll count that as a point for evolution. But the problem I'm having,
done that math. I've done that math.
No, I watched your video. I watched your.
Yeah, it's
I watched your video
30,000 times, off by 5 orders of magnitude.
No, I did. I watched your video, but you told me
that.
you didn't account for the mutations in that.
No, I did.
Okay, well then I'll rewatch it.
That's the point, is that in the creation model, you can't do it. You need to do it via recombination, because if you do it via mutation, the lineages diverge from each other and you lose the hierarchical pattern. So in the creation model, you must account for that variation almost entirely, not strictly, but over 90 some odd percent. Nathaniel Jeanson has the actual number, but I think it's like 98%. It has to be due to recombination. And that's not my number. That's Nathaniel Jeanson's number. Okay.
And I accept that. But remember,
that number requires that number requires the rate of recombination be 30,000 times faster than what we observe.
Okay, well, I have not looked into that, so I can't talk about that. And I will. Okay. And if what you're saying is true, I will grant the point against my young Earth position. It still won't be a point toward evolution, but it'll be a point against the young Earth position, right? Like I'm counting it as a point against mine. Now, but what I'm talking about with the demonstration, remember, we weren't going with one kind. We were going with 3 different kinds. So what I'm asking is, why can't my starting point be the same as your starting point when you said common ancestry. And this is the problem I'm having with.
I gave your starting point. Your starting point was a perfect nested hierarchy. Why would you want a different starting point?
Well, but I'm saying it's
the maximally generous starting point.
Well, I don't know that it's the maximally generous starting point. Okay,
because in the common ancestry, you didn't have a perfect nested hierarchy. You saw some variation in there. In the common design starting point in my model, it was a perfect nested hierarchy where out of the 15 sites, 5 were shared by all 3, 5 were shared by 2 and then different in 1, and then 5 were different in all 3. That is a perfect nested hierarchy.
But
you can't get
but
that.
it
Focusing a bit too much on the hierarchy.
Starting point. What's
I
that?
was going to say that assumes that you know what we were starting with to begin with, Mr. Dan. I really appreciate that.
You can pick any starting point you want.
But that's why I'm saying, Dr. Dan, that's why I'm saying, can I pick your starting point? Can I pick your starting point? You took 5 that were you copied and pasted, right? And you kept those and you generated variation
because they shared common ancestry, right? Because they happened in the past when they were common ancestors.
And what I'm saying is I would like to take that starting point and say they didn't share common ancestry, but they shared common genes. They didn't those genes didn't have a common history.
Right.
But the genetics shared commonality without sharing common history.
So how do you get the existing variation if they don't share common ancestry, right?
Right.
They were separately created with very few variants at creation. That is a necessity. We agree on that. How do you get the existing variation? How do you get the existing diversity?
Well, you didn't have to get them to get the existing diversity because they didn't start out together. We were doing human, chimp, gorilla. So they didn't need to get to any.
So you want them to start randomly then with just random diversity in each lineage? Because I'll tell you,
I wanted them to start with your starting point. I wanted them to start with your starting point.
But my starting point was probabilistically determined based on a series of nested mutations.
But that's what, but the whole point of this, Dr. Dan, is to show you why I'm having a problem with the calculations for separate ancestry. It's because you are assuming a starting point. You're still not admitting that, and neither is the paper, but there is a starting point.
About this. We talked about this the other day, that when you say assume, you're saying as a basis for my hypothesis, this is the starting point, right? So in my model, for separate ancestry, I assume they were created separately with the appearance of a nested hierarchical pattern. With the common ancestry, I assume they share a common ancestor and accumulate subsets of mutations in their common ancestors.
Yeah, just FYI, like if you went with Dr. Dan's starting point, you would be going with a shared common ancestor. So it would no longer be separate ancestry. But Dr. Dan's starting point for the separate creation model is the one that gives you the closest to the results that we see. Like if you used any other starting point, you could try it yourself. Input any possible other starting point, you're going to get a worse result.
It'll be worse. Dan. Yes. Thank you, Grayson. Yes, exactly.
But you guys are that's the point I'm trying to make is you guys are starting with something, right? So you're saying in this paper, they don't have they're agnostic at the starting point, but it's not agnostic at the starting point.
It is. It literally is. It literally is. There's no, like in my model, you can say, Dan, what sequences did you start with for run number 4 of your separate ancestry model? And I can say, here are the sequences we started with, right?
But that just points out the assumptions that you made, Mr. Dan. That's the point.
No, wait. I was making a very narrow point, and my point is this. In the paper, you cannot say what sequences did they start with. You can't, right? You can't do that. They didn't start with sequences and work forward. They started with existing diversity and work backwards. It's a different way of doing it. Now, my model is simplified, right? So I'm saying, let's start with a thing and work forwards. And you can say that there are assumptions built into my starting states, which is correct. There are, because we're hypothesis testing, right? So each hypothesis has assumptions that are inherent to that hypothesis, right? That's how hypotheses work. If you think that my model was wrong or uncharitable in some way, right? By saying, I'm going to start the separate ancestry model with a perfectly nested hierarchy and perturb it as mutations occur. If you think that is an uncharitable way to do it, suggest to me a better way to do it. Now, I can't just do the same thing I did for common ancestry, because then you're going to get, like, then it's a common ancestry model, right? Where the lineages share nested sets of mutations, right? That's common ancestry. So if you would prefer an identical starting state or a random starting state, whatever, we can redo the simulation with whatever starting state you want. But I'm telling you, the way I did it was the maximally charitable starting state for the separate ancestry position.
If it was maximally charitable and you said I can choose any starting state, I'm saying I want to choose the starting state that you chose.
But
then you're choosing a shared common ancestry.
No, that's your assumption that that's common.
No, it's not. That's just how. Look, we have a level
it
of
is something you're imposing onto the data. I was going to say
we have a level of variation that exists in the present, and at creation we agree that you can only have in each created kind a small subset of that variation, right?
But you're comparing.
Hold on, but we have to know what the genes were, what their functions are, and everything from the start. We don't know every single thing that was supposed to be there from the start. That's what you're assuming.
Can I try this?
How many alleles can you get at the start? Tell me a number. How many alleles can you get at the start with a population of either 2 or 7, depending on the species, coming off the arc? What is the maximum number of alleles you can get? Tell me a number.
It depends on the actual.
It literally
yes, it does. It depends on the genomes that we had to start with.
No, it doesn't.
Yes, it does.
It depends on the number of things that get off the arc. That is it. If there are 7
but how do you know what the starting point is
for
those animals that got
alleles? If you have 7 individuals of a kind that get off the arc, you have a maximum of 14 alleles, right? Yes or no?
Wait, if you have...
This is a question for Pigs Can Fly. My understanding, I'm not a Bible scholar, my understanding of Genesis is that either, depending on whether they're clean or unclean or whatever, either 2 or 7 of each kind got off the ark, right?
Yeah, but you're assuming that the variability is not right. Hold on, stop, Mr. Dillon. Please let me respond before you continue, because you're asking me a question and trying to respond to you.
I just need a number.
No, you don't. You need an actual response instead of something that's...
How many alleles can you have in 7 diploid individuals? How many distinct alleles can you have?
Yeah, you have the 14. However, we don't know what the maximum genome size looked like, how many children they had. We don't know anything about that stuff.
Doesn't matter.
You're assuming so much stuff with the way you're asking the question.
Okay, yeah, I agree with what you're saying, Dr. Dan. Okay, right. There's a certain number of alleles that they can have. And now, I mean, there might be...
And you can make them in whatever pattern you want, similar, different. Right. It doesn't matter, because the point is you only have this many, and you need to generate this many.
If it's 2 animals getting off the ark, then you have 4 possible alleles, right?
Possible alleles.
Okay. Now,
for many loci, you need to generate thousands.
Exactly. Now, we're on the same page with that. We're on the same...
Do we agree that the main way that's going to happen is mutation?
Okay, well, we're going to have recombinations.
But the recombination rate is 1 crossover per arm per chromosome, per meiosis. So we can calculate the mutation, the recombination rate. I've done that math. And it's, again, several orders of magnitude too slow to generate the variation.
Okay, but remember what you're starting with. So, but in the paper that we're discussing, it is not talking about getting the variation in one kind. It's comparing between human, chimp, and gorilla, which we say are separate. So we don't have a starting point. Now, if we were able to say, you know, what they were when they got off the flood, then we could calculate, you know, the differences or whatever. But it's the starting point is what we don't know. We don't know. We know the number of alleles that can come about by recombination. And I mean, generally we can calculate, and mutation. But
you can
don't know for human, chimp, and gorilla is what they actually started with. How many similarities did they start with?
And what I'm trying to convey is that the model, the way they did their calculations is that the calculations converged on the maximally charitable starting point for each model.
But there's no maximally charitable starting point for our pattern, because we could, this is what I'm going to say, there's not a scientific starting point.
We have the existing variation, right? We have we have A, we have the arrow, and we have B, right? And you're saying, well, what was A? And I'm saying they didn't tell you A. They didn't start with the defined A. We know B, we know the ending states, and we know what the arrow looks like. So they're saying, given those two things, what is, if we had the best possible A, what's the likelihood you get from separate A's to all the B's, right? They didn't define those A's. They just used a model that found the best ones.
Are you two trying to say that somehow these other alleles that were not at, like, you read that there were 4 options for alleles at that location if there's only 2 animals. So are you saying that they had all the other alleles at different locations in the genome, and it's like somehow through some mechanism all of those alleles migrated to the locations that they are currently in all the different ancestors? Are you trying to say genetics worked differently in the past?
No.
Well,
then I don't understand what you're saying.
What's going on here? No, what's going on here? Go ahead.
So there's, I already posted this in the chat, and it was about there's a dependency graph model of life that was presented by Dr. Rob Stadler and his discussion. Yeah, I understand that you don't like that. But yeah, exactly. Western, you're right. And so that was presenting that essentially it's very similar to what a software program would utilize. There's a large database that already has all the existing codes that are, for whatever particular task or function. And so they would just pull from that to try to start moving forward and building on their own personal programming, whatever they're trying to do at the time. Now, we're trying to figure out why Dan doesn't want to be actually charitable in that sense to the designer and allow him to just be that person who picks and chooses this particular gene as opposed to this particular gene with this particular function at this point in time to utilize, instead of assuming that there had to be common ancestry, which is what Rebecca's trying to point out. So that's what I was trying to get across before, and I'm not sure if you actually heard that part whenever you were coming in, Dr. Dan. But it's kind of strange. You knew about the paper, but why haven't you been talking about it at all?
I haven't been talking about it because it's exactly the kind of post hoc thing that Rebecca you and Rebecca.
So this was from over 5 years ago. How was it post hoc?
No, it's what I'm saying is the way that model works is it builds the conclusion into the model. And if you want, we can
Wait a second. No, it doesn't.
If you want, we can pull up the paper and go through it.
All it's doing is it's comparing the genes that are actually functional and seeing why this would not work if we're using a designer from the start. It's just like you doing with the separate entry.
No,
you're assuming that it doesn't have to have a designer. And then you're saying, well, this is the preferred model this is the case.
Is a dependency graph probabilistic or deterministic?
Well, it would depend on the actual designer, right, of course.
It's deterministic, right?
It can't be deterministic in the sense we're talking about. It's not saying that it's because of physics and chemistry or something like that. It's saying determined according to what the designer would put in place. We don't know what that would be because we're not the designer in question. You see what I'm saying?
Right, but that's not what deterministic means in this context. Do you understand that?
Can you explain it to me then?
Yeah, it means the outcomes that occur, right? When you do phylogenetics, right, we talk about deterministic versus probabilistic models, where probabilistic generates a range of outcomes, and given the parameters, you evaluate which ones are most likely, right? So you generate a probability distribution, and you converge on the most likely outcomes. When you have a deterministic model, no matter how, let me go back a step. When you do a probabilistic method, if you do it 10 times, you're going to get 10 very similar but slightly different answers. When you do a deterministic model, you're going to get the same answer each time, right?
Wait, well, hold on. So the deterministic model requires...
Hang on. There are methods of phylogenetics, like neighbor joining is one of these methods, where you can use the same data set, do it 10,000 times for neighbor joining, you're going to get the same tree 10,000 times. But if you put the same data set into a maximum likelihood model or a Bayesian model or whatever, you're going to get very similar but different phylogenies each time, right? So that's the difference between a probabilistic and a deterministic methodology. And probabilistic methodologies are generally considered much more robust because they account for the uncertainty in your data set and the incompleteness of your data set, right? Whereas deterministic models have a false sense of confidence built in because you only get one answer each time, no matter how many times you get one answer, no matter how many times you do it for a given data set. And this dependency graph stuff, and this is, for anyone curious, there's a really long thread on this on the Peaceful Science forum. Peaceful Science, spelled exactly like you just Google it. There's long threads on this on there, going through this like line by line. But it is a deterministic process rather than a probabilistic one, and it builds in a false sense of confidence. So essentially what Ewart is doing is overfitting the data, which is a no-no.
No, I mean, we obviously know that there's assumptions in both cases, right? And it's actually stated in both papers what those assumptions are.
Now you're just swiping aside the substantive criticism. Do you see what just happened there, though? I made an actual very specific critique of the methodology, and you were just like, both sides. Do you think my critique is valid or not? And if not, why not?
No, I do not. I don't think it actually characterizes the situation properly. So obviously, because you're not pointing out the actual assumptions that are in your own paper, first of all.
Whatever. Pull up the blog post.
Okay.
Pull it up. I'll go through it with you right now. Pull it up. I'll show you. You're not gonna do it? Okay. Fair enough.
Do you want to do that, Matt?
Again, I'm on a phone. I'm on a cell phone. And
Yeah.
I've been
okay.
the whole time. But I
How
posted the papers in the
do
chat
that?
there so that they can take a look at what we're talking about.
Yeah, and I mean, Dr. Dan, I'm also willing to be reasonable on that. Like I'm not following this argument because I haven't looked into that. But I do just I wanted to answer Grayson's question about what we're doing with the separate ancestry thing and answer. I think we are conflating two different issues, because if we're talking about variation of alleles, like what we see starting with in alleles of the same kind, right?
Mm-hmm.
Then
Mm-hmm.
we can go back and kind of like say we know at the flood, basically if we're going with the flood model, then there's only a certain potential, right?
Mm-hmm.
But
Yeah.
but the critique that I'm making about the paper that compares the separate ancestry and common ancestry with regard to primates is that we're not starting with that same thing, because we don't need to get we don't have to get to a certain variation. Okay.
You do that because you have to get to existing variation.
So are you, but then am I misunderstanding? Because from what I'm understanding,
we all need to arrive at, to borrow a phrase, we all have our limitations, right? We all are constrained by what currently exists in the present that we've documented.
Right.
So no matter what your model of origins is, you have to arrive at the current level of biodiversity that we have documented.
Yes.
And we have a specific set of processes that we have very specifically documented to get us there. And we've talked about this, and you're not proposing like ongoing tinkering or vastly different processes operating 4,000 years ago compared to now, right? So we know what the processes are to get us from A to B.
Right.
We know what B is. So what we're trying to do, if I understand your critique, is that we're not giving you the correct A, right? We're not giving you the right starting point that gets us to B given the processes. Is that fair?
Yeah, exactly. Like B
is
what we see now. B is what we see now.
B is what we see now.
And we're
I'm
trying
not
to get
giving you the right A, right? I'm not
Yes.
giving you the right A that allows
Yes.
your model to get us there. What I'm trying to convey is in the little simulation that we did together the other night, I'm giving you the best possible starting point for your position by building in the current pattern. And then in that paper, they do even better than me by saying, we're not going to pick a starting state. We're going to let the data tell us what starting state gets us the closest.
But how can you compare starting states if you're not assuming a starting state?
By
What I
letting
mean
the
is
data determine the best possible starting state.
Part of it too, if I'm understanding it correctly, is that like a lot of it depends on like how many steps it would need to take for like sequence A to transmutate into sequence B, right? Like you can have different things like that happen, like where, for instance, like A mutates to T, A to T, A to T, A to T, A to T, and eventually end with an A, right?
Right.
But you can also just have an event where something that was a T mutates into an A, right? And this, right? And the kind of thing that can happen, right, is both of these are like plausible, right? But it's very unlikely that you have this like single nucleotide, that's mutating every generation between A and T, right? God knows how many times, versus one single event of, like a T being mutated into an A. Right.
Exactly.
So there's certain events like that when you're doing these computations, right? You can have the same ending sequence, right? But certain processes just probabilistically, given something about the rates at which they occur, like that they're observed to occur at, how likely is it that this process gave you the sequence that started with an A versus this one that started with an A? And the algorithm is taking charitably the one that has the highest likelihood.
Thank you. Yes, exactly. Thank you.
Okay, so and look, I'm not trying to give you guys a hard time unnecessarily, okay? Like I will just admit up front, I could be massively misunderstanding this, okay? So I am open to being shown what I'm missing, but I still don't feel like you guys are listening to me about the starting point.
State would you like?
Because what the question that I would like answered is if like I'm saying, look, I cannot know the starting point for human, chimp, gorilla. I can't know that starting point. I don't know which genes they had in common, even though they were created separately. They may share common genes, okay, because they're trying to get the common functions. So what I would like to choose, since I know Dr. Dan's starting point for that demonstration leads us to the where we are today, I'm saying, why can't I choose that as my starting point for separate ancestry?
You can, but that makes you an evolutionist.
Can I try this one?
Well, it doesn't,
Rebecca. Dan's model does not start with 3 different genomes. His starting point is one genome that then mutates and evolves into 3 different ones. So you cannot that's the reason why you cannot use Dan's starting point, because his starting point is not 3 separate genomes, it's one genome. So if you used his starting point, that would be common ancestry.
Okay. Now, okay. But it's not going all the way back, right? Like it's okay.
Yeah, okay, but it's not supposed to.
Okay, hold on. You guys might be having I might be having a breakthrough. Okay, you might have finally helped me understand. Okay, so just hang on. I'm not sure I can declare it right now, but let me think on it. Matt, do you have any objection to this? Are you following this argument? Because I know you may not have been part of all this. So
no, I mean, I still feel like there's a certain starting point they have to assume every single time. It doesn't really matter, and it's always arbitrary, even if they want to say it's not. Ultimately,
can I ask Pigs Can Fly a quick question? Is there any starting point that you feel like separate ancestry, like any possible combination of genes and alleles and whatever, any potential starting point that could result in our current diversity today?
You mean through common ancestry, you said?
No, through separate ancestry. Like you're saying that, oh, there's assumptions about the starting point. I'm saying, like, out of every single mathematically possible combination of nucleotides, is there any possible starting point that could result in today's diversity patterns?
I mean, if we have all of the different mechanisms that are actually categorized and taken into account, I think we can get a proper model. Yeah, but that's what I was mentioning before about the mutated genome decay. I don't think that was even taken into account to any of the things that we're talking about. And of course,
Hang on.
Dr. Dan
Again, I
assumed,
want to be clear. No,
Dr. Dan
just
assumed,
I
here
want to be
we go
clear
again. We're going to keep.
about decay. I
Listen,
literally
Dr. Dan,
told
I've sat here for literally over 20 minutes just listening to you. I'm sorry, sir, but I just have to put it
I
straight
mean,
to
he
you. Okay?
addressed your point already.
I did address your point in the
Yeah,
No,
he
you did
directly
not.
explained the
You assumed-
mutator genome decay despite sustained fitness gains paper. He explained the survival of the flattest. Like, he did go through that explanation.
I did address that.
Okay, so now you guys are saying Dr. Dan's starting point is complete equal genome, like one genome is the starting point. But what I remember is that we took 5 of 15, right? Like, and I know this is greatly oversimplified. And what I remember from this, basically the the paper that I'm reading is it's, are we really starting from a like completely the same genome?
They're not
Are you
starting
saying
from
that?
any genome in the paper. They don't pick a
Right.
starting state in the
Yeah,
paper.
Dan's simulation models has a starting point, but the paper itself does not
Paper does
assume
not have a
different
starting
points.
Okay, so
It doesn't have a fit. We should say it doesn't have a
Right.
fixed starting point. If you dug into the models, if you ran their models, you would converge at an optimal starting point for each model.
Okay, so now let's go back to your demonstration. Why can't I have the 5 alleles, the 5 nucleotides
Right,
out
5
of
and 5 and
15,
5, like I did,
right? Yeah, and we're greatly oversimplifying. 15 is not going to be
because it's
accurate,
haploid and whatever. Yeah, right.
but why can't I have 5 that are the same
Out of 3 that I made.
instead of 3? Because basically, you know what I mean? Like you did the nested hierarchy and
and then I compressed it and varied the rest, right?
Right. But
why
why
can't
can't I have the same starting point as you? Because I don't see how that starting point is common design.
Because you have to generate the existing variation in each lineage through mutation. In the common ancestry model, some of that variation occurs not in humans or chimps or gorillas, but in the common ancestor of all 3. Some of that variation occurs in the common ancestor of humans and chimps, and some of that variation occurs independently in each lineage. In the common design model, the vast majority of that variation must occur independently in each lineage. So I was actually being generous doing 12 out of 15. That was, 80%? That's actually generous given the amount of variation you need to generate in each lineage compared to the starting variation in each lineage.
But how do you know the starting variation in each lineage?
Because you're set by the number of things that exist at creation or get off the ark, whatever thing you want to use.
But how do you know how many things are set at creation in the ark? But we're going between 3 items, not one kind.
If you have, let's use humans, right? Let's use humans, right? So you've got Noah and his wife, and they're going to have Noah's 3 sons, right? So in Noah and Noah's wife, you have 2 individuals, which is 4 alleles. You can have 4 variants of each locus in the genome, right? We agree on that. And then in the wives, you have an additional 6 between the 3 of them. We're assuming they're completely unrelated, right?
Yes.
So that means the human family getting off the ark has a maximum genetic diversity of 10 alleles for each locus in the genome.
Okay.
Right? Okay.
Yes.
For many parts of the genome, you need to have at present over 1,000 variants. So you need to get from 10 to 1,000. And that's not just for humans, that's for everything that gets off the ark. So how are we going to do that? It has to be via mutations that occur independently in each lineage. That's why out of that perfectly nested hierarchy, I said, okay, let's compress it. And then we're going to take 80% of it and mutate it.
Right. But remember, that is a separate argument because in this paper and in the demonstration, we're not trying to get the variation within humans. We're trying to calculate the variation between chimps, gorillas.
But you can't do those two things independently. You can't generate, like if we wanted to, we could just say we start with the nested hierarchy and it stays the same. Great. But then you're going to have no variation within each lineage, right?
But what I'm saying is, okay, let's do it. You've got 10 alleles for the humans, right? Is that what you said? 10 alleles?
It was 10. Yeah, it would have been 10
now,
getting off the arc, right? Your maximum number of alleles is 10 for each. Yeah, for each.
And then for chimps, you've got 4.
4.
Okay.
Yeah.
For gorillas, you've got 4.
4. And you have to generate 1,000
now,
say, in each lineage.
How do you know which similarities? How do you know what the gorilla? You know it only has 4 alleles for whatever thing, right? But you don't know what it shares genetically with humans and chimps.
And my assumption was that comparison, that pattern of similarity was a perfect nested hierarchy. That was my assumption.
Okay. A perfect nested hierarchy, but
when they stepped off the arc. Perfect.
Because that makes it the most probable to reflect today's current diversity.
Maintain the nested hierarchy that we speak. Yeah.
But the nested hierarchy is the evolutionary scenario, right? I mean,
but the nested hierarchy is what exists in the present. So we both have to get to that from our different starting points, right? That's the point.
Right. Yes. Okay. Yes. So you're saying point B is a perfect nested hierarchy.
No, it's far from perfect, but it is a nested hierarchy. Yeah.
Okay. But again, like, why can't I choose your same starting point? How is that starting with all the same genomes?
When you say do you want 5 nucleotides shared by each lineage?
I want
the 5 that are the same across all 3.
The 5 that you did. I don't remember exactly what you did on your thing.
It doesn't matter.
I think you did 5
that were the same. Okay, but the point is, if we start that way, I mean, sure, we could start that way, but the point is, that was the end result for me, right? That was saying at the end of the process, because the beginning is a common ancestor that's necessarily the same because it's one common ancestor. They start identical. So are you saying you want to start identical and have mutations accumulate? Because we could do that.
That would mean that Noah and his family had identical genomes with all the other animals.
Well, that's what I'm trying to figure out. Look, if you're right, then I'll grant the point.
Rebecca, I think I understand what you're saying. I think I understand what you're saying. Okay, let me try to see if I'm correct on I think it just clicked for me. So what I did is say for the common ancestry simulation, I said they all start the same. And then they share that history for some amount of time, right? But then 2 lineages diverge. So now they have a different history. And then 2 more lineages diverge. They have a different history. And so you're saying, why can't you start the same? Like I assumed they started, right? In mine, it would have been like, oh, geez, or whatever, right? Whatever it was, right? And then, but the difference is, and here's why we can totally do that. I would love to just pull up the random sequence generator and do that. But the result of that will be gibberish for the common design simulation. And the reason that's the case is because you still need to generate the variation. So that means you can start identical, but then you still need to generate all those variants over the course of their evolutionary history. And instead of sharing a subset of those variants as they mutate, they're all going to accumulate mutations independently.
Okay, but like the thing that I think the difference here, though, and you can tell me if I'm wrong, is that like I only have to get the variance to generate within the kind.
And make a nested hierarchy. And make a nested hierarchy. There's 2 constraints here: existing level of diversity and a nested hierarchical pattern. You have to get both things.
Okay, wait. Say it one more time for me. You
need to get the existing level of diversity.
Yes.
And you need to get the existing nested hierarchical pattern.
Okay.
Right? So you can generate the existing diversity. If you have an identical starting point, it's trivially easy to generate the existing diversity. But because each lineage again, not my assumption, your model, right? Your hypothesis, right?
Right.
Each lineage is doing its own thing. So those mutations are going to be uncorrelated with each other. You won't get a nested hierarchical pattern.
Well, let's look.
I was going to say that, okay, so again, you're assuming that these are not pre-existing genes that are already there.
That's wrong.
And that's where, yeah, I think like maybe the difference here is that you guys have to get all the variation from the various kinds. Okay, I admit I have to get the variation within the kind, but you guys in your model, you have to get all the variation between all the kinds, but I'm not limited on that.
You. Well, you are, because the existing pattern exists, right? So your starting point has to end up at reality, right?
But that's why I pointed out the dependency model, right? The dependency graph model, because that's supposed to explain the very thing you're talking about. And it was actually highly favored above
Let me say it a different way. The dependency graph model has deterministic rather than probabilistic mutations. Let me put it that way.
Okay, what does that have to do with the fact that it's
Because
highly
mutations
favored?
are probabilistic,
Assuming
and we can show that from an experiment from 1942. Mutations are not deterministic, right?
I didn't say that the mutations themselves are. So here's the thing. So again, you are assuming that these genes are being built up from the bottom up. They are not existent, and that they are slowly coming into existence over time by developing mutations.
Hey, Pigs Can Fly, can I just ask a quick question, just so I'm trying to remember? There's a bunch of people you encounter in these various things. Are you the guy who doesn't think that dogs and wolves share a common ancestor? Is that you?
What? No, I didn't say that.
I believe that is Bubblegum Gun.
Oh,
yeah, Bubblegum Gun.
Oh my God, I'm so
Bubblegum Gun.
I remember him saying that because I actually got him. I got on him about that. I was like, "What are you talking about?"
Reputation with my confusion of different people. I apologize. Okay,
it's okay.
Just making sure. Okay, cool. Just cool.
Okay, guys, you know what? I do have to work tomorrow, so I'm going to have to, and I'm sure you guys do too. Look, but Grayson wanted to show something. He has evidence that he wants to present. Grayson, do you think you can do it in like 5 minutes? Because otherwise,
I'll go. Okay, I'll go. Grayson, thank you for pinging me, or like just mentioning me earlier. And Pigs, thanks. Rebecca, thank you for having me on. It's always a pleasure.
It's a good conversation.
Thank you, guys. I mean, I really don't want the conversation to end because this is really good. But I'm going to tell you, I am going to think about what you're saying here. I will rewatch this. I will read the paper you sent me. I will think about all of this. Okay. So I like that we have this conversation recorded because I can go back and try to understand what's going on. Okay. So thank you very much for patiently working with me and thinking through what I'm saying. So appreciate you guys.
All right.
Yeah. Thanks for having us on. So my point was I think that the genetic evidence that we just went over is sufficient. Even if we never had a single fossil ever form on Earth, we would be able to conclude evolution happens based on the genetics that we just went over. But it goes beyond that because we do have fossils, and specifically the pattern of the fossil record matches the pattern of genetic similarities that we've been talking about with the nested hierarchies shown in phylogenetics. So I will just share the screen here. And this is from data in insects. If you could get the screen on. So basically, they look. Sorry, you're muted, Rebecca, but I'm sharing the screen currently. But basically, they're looking. You're still
I'm trying to get it on, but it's.
I'm following what you're saying, Grayson. Go ahead.
Okay, so from left to right here, this is just like a phylogenetic tree. Basically, this group over here, Diptera, is the most genetically similar to the groups that are closest to it over here, these two groups. And then these groups over here are most genetically similar to these. So this is just, looking at the patterns of genetic similarity, right? And now you can do a phylogenetic analysis of that. But the crazy thing is that this is representing the fossil record. So you can see that the order that we find these things in the fossil record matches with the order of their genetic similarity. Now, water from a flood cannot sort things according to nested hierarchical patterns of genetic similarity. And additionally, you also see that these fossils, the thickness of them is basically how many different varieties of fossils are found in each one of these families in the fossil record. So you can see here that for each one of these different types, basically, you get this pattern where you have a lot of different varieties in the more recent rocks. And then as you go down in the rock layers, the amount of diversity goes down, which would be exactly what you would expect with evolution. But the pattern of which they appear also reflects the pattern of genetics. So we see here, this one over here appears over at this level. And the one that it's the very farthest from, like the most distant genetic relative, appears way down earlier than everything else. Okay, so you can literally just map how similar they are according to their genetics, and then you can predict the order at which they are found in the fossil record. A flood cannot do this. That is what I consider to be the strongest evidence.
For the creatures that are down, I mean, like the creatures that we're finding in the fossil record.
So these are all these are currently let me go back to this.
I think it's so by are you going by morphology, not genetics?
Yeah, that's what it sounds like. And
so this is,
I was going to say, how do you make the correlation between what's actually genetically linked? Because these are all fossils,
yeah, I'm
right?
trying to explain it right now, actually. So you can sequence the genetics for all the currently living species, right? They are in a certain family, right? That it's Linnaean taxonomy that predates evolution, right? You can group things together. And in the fossil record, you can tell, like, hey, this is a fly or this is a grasshopper. You know, you can tell based on the fossils which family that those fossils belong to. You can genetically sequence the living members of those families to compare the genetic similarities of the extant species of those families. And then you can find extinct species in those same families in the fossil record.
If I may, I just wanted to point this out. So, for instance, I have a cousin, right? He's much larger than me, and I just want to point that out. So, if we were both buried at the same time, you would not have any idea like how closely related we were actually in blood or genetics and things like that. And assuming that we in a flood got this the disposition and put somewhere else, right? There's different biomes in this situation. We have no idea where these creatures were located and how they would have been put down, assuming that there was a flood, right? So I'm not sure you can really make the correlation that you're making here. I'm just trying to clarify. It sounds like there's an assumption here that these are already genetically linked based upon that, and that they were already in certain locations throughout history, right? You're assuming a certain time period. You're assuming multiple things here at the same time. Now, just to be clear, I understand your argument. It's actually pretty strong if you have nothing else to go off of. But the issue I'm saying here is that it does also kind of jump past certain points that could actually defeat that. I wonder if that's being represented there.
So you're classifying the fossils according to what family they're in, right? Are they a fly? Are they a butterfly? Like, are they a spider or a dragonfly or whatever? Like, you can look at a fossil and tell, hey, you and your cousin are both in the hominid family. You're both humans of the same species, even if he's just a little bit bigger. You can still tell you're the same species because you have the same morphology. Like, you can look at a fossil and tell if it's a grasshopper or a dragonfly. Yes, you agree? So you can tell if these fossils are in the same family or not. So it's not like you're just,
yes.
I mean, yeah. So then you can just say, look, and this is for all places around the earth. Like, this is like the total amount of fossil species within each of these families. So they're saying, like, here for like for dragonflies up here, when you look at the fossils, you see a lot of variety up here. There's a little bottleneck where they're not finding as much variety, and then you find a lot more variety of dragonflies down here. But the general pattern is this widening pattern that we see across all of these. So
right, but that doesn't actually speak to them being. Listen, that doesn't actually speak to what I think you're trying to present here. So ultimately, if you think about this, right, it's just saying that there were certain amounts that were deposited at certain layers, right? Not necessarily that there was these vast time periods that were extending their ability to reproduce, things like that, right?
I don't care about the time period. I don't care about the millions of years. I'm just saying this is the pattern in the fossil record, in the rock layers,
Right.
we do observe a certain pattern of distribution.
Okay.
And that pattern of distribution.
Do you think that contradicts our view? Because to me,
yes.
So far what I'm seeing, it doesn't really contradict my view. Meaning,
so there's
like,
not
I would expect diversity from the time of the flood. Like, if those fossils were laid down in the flood, I would expect things have diversified since then. So I would expect the same kind of cone.
Yeah, you're just not expecting them to go back to a common ancestor. I see.
Yeah.
So I see that the cone is sure it predicts both, but I'm saying that the layering, like the order at which we're finding these things, is clearly correlated with currently measured genetic similarities. Like two things that are like we see on the far right and on the far left of this graph, the two furthest genetically similar, you can also see that's where we see a disparaging amount of the ordering that they appear in our fossil record. The ones on the left appear earlier than the ones on the right, or at least, deeper down in the layers if you don't want to use time and order or
right.
So we do see that pattern, and that pattern, you were talking about mechanisms before. You were saying like, oh, I just don't see the mechanism. Well, that's what I'm saying, is that there's no mechanism by which a flood could sort these things in this way. Ecological zonation that you were talking about doesn't work for a variety of other reasons, but I would just bring up the order that pollen is found in the fossil record debunks ecological zonation. See, pollen is found on every biome on Earth. If there are flowering plants on the surface of Earth anywhere in any appreciable amount, you will find pollen everywhere. Right now, today, we can find pollen at the bottom of the ocean, on Antarctica, in the deepest deserts, whatever. And in the fossil record, we see layers that do have pollen and layers that do not have pollen. I see that Rebecca has slides prepped for pollen that she maybe wants to get to. So I'd be happy to hear an explanation for that, because I've never heard an adequate one. Do you hear what I'm saying? No PC flag with pollen?
Yeah, you know what? Actually, it's weird, Grayson, because I actually started this show talking about pollen because somebody brought that challenge last time. But I'm not going to bore you guys by going through it. Look, if you want to know my answer on pollen, go back to the beginning of this video later, okay? But look, guys, Grayson, I'll think about what you said, but let me just say one thing about that chart. The chart, at every layer, I'm seeing things that are very similar to things that are on Earth today, right? Yeah. So it's like there's like fully formed insects, fully formed sea creatures that are there throughout all those layers.
Right, so, and the ones that were older too, like the oldest ones there, those were silverfish. I actually used to see those on the docks and piers when I worked on the submarine, and I was like, well, that's pretty crazy that those things were supposedly here for so long. But anyway,
right. Well, I mean, I feel like if you understand how evolution works, that wouldn't really be surprising, right? Because things evolve to optimize for a certain niche in their environment.
Well, and
if that niche in the environment is not changing for millions of years, then why would they change if it's working so well for them for those millions of years? So if you understand how evolution works,
optimization, though, Jason, that's what I wanted to ask. Is like, so there's always this assumption that there's some sort of optimization.
It's not an assumption. It's also directly measured, like fitness landscapes, distribution of fitness effects. It's in Dan and Zach's paper that they provided you.
But here's the thing. So optimization itself, doesn't that also require a standard to be in place just for you to do so? Am I not missing something with that?
Yeah, the standard would be the interaction with the environment.
Okay, but don't you understand the issue with that? So who would be determining that this is the best case scenario? You're not understanding what I'm saying with that?
The interaction with the environment. It's very clear cut. It's really simple to understand, right?
No, it's not, because here's the issue, right?
It is, though. If you have a creature that has a lot of hair in the desert versus a creature that doesn't have a lot of hair, one of them is going to be more optimized for that environment. Clearly,
I mean, camels are covered in hair and they live in the desert. That has nothing to do with it. The whole point
is that everything to do with it. It is the interaction with the environment that provides the standard by which these things optimize. That's clear, simple, easy to understand.
You're not getting the issue. So optimization requires a standard. Do you see what I'm talking about?
Yeah, the standard for the optimization
we can judge that is optimized. Do you see what I'm saying? You and I,
I'm explaining it very simply to you. I'm explaining it extremely simply. The standard by which these things are weighed against is the environment in which they live. If they are successful in that environment, then that is more optimized than ones that are not successful in that environment. It's literally just how do they interact with the environment that they find themselves in.
All it's saying is that things that are simply are is what you're telling me, and that's not an actual answer.
No, that's not what I'm
to the
saying. That's
question.
a straw man. Don't put words in my mouth,
That
sir.
is what
I
it's
don't
that's
appreciate
basically
what you're
you just
doing.
said to me.
That is not
Okay,
what I just
guys,
said to you.
I
Everyone knows
really-
that is not what I just said, and that is not an accurate way to summarize what I
You said
just
that
said. To you.
because things exist that live in those environments and they are optimized because they exist in that environment. Therefore, that's the most optimal, right? That's what you're telling me.
That's not what I
That is
said.
just
That's
is
clearly not
at-
what I said. I don't know why you're trying. I don't know if you're intentionally misunderstanding me or what. I feel like I'm being very exceedingly clear on this issue. You have a amount of diversity, right? None of them is preordained to be more optimal. You have all these different varieties. You stick them all in one single environment. And that environment, the way that they interact with that environment, is what dictates which of those different forms is the most optimal.
as I mentioned
very
before.
clear.
That's
Rebecca, you
exactly what I
understand what I'm
just
saying,
told
right?
you.
Okay, let Matt say 1 minute, then Grayson can respond 1 minute, and then we'll close it because I got to go to bed.
Okay, but really quick, Rebecca, you do understand what I'm saying, or am I not making myself clear?
I would like to not carry. How about let's pause the conversation?
This is actually, kind of not really that helpful. I apologize, Grayson.
No.
But just to be clear, I just thought that I was not, being facetious or anything like that. I'm just trying to clarify. It sounds like you're saying reality is reality when I hear that argument.
I'm really not. If we can talk after this, if there's somewhere where we can continue talking after this stream, I'm happy to talk with you.
You guys can keep
I wish I could because I only have like 8% of my battery.
StreamYard here. Once I shut off the thing, you guys can keep talking. But yes, I'm happy to pick up that conversation another time. Do you guys want to make a final point each and then I'll close it? Or do you want to just pause it and say, okay, good conversation, pause.
Yeah, we can just pause it.
Okay. Awesome. All right. Well, thanks everybody for watching. Appreciate everybody who was here. And Creation Myth says new stream. So I think, okay. So he's saying I'm on summer break. Someone make a new stream. So if any of you guys want to do a new stream, then go for it. Okay. Thanks for coming on and see you next time.
No problem.